Ladies and gentlemen, a very good morning to all of you and welcome to the 6th International Conference on Molecular Biology and Biotechnology 2023. My name is Yik Ling and it is my honor and absolute pleasure to be your MC of the day. Before we begin, allow me to make a few administrative announcements to ensure that today's event runs smoothly. Please mute your video and audio. You will be given instruction if you need to turn on your video. If you leave the room and want to rejoin, you may click on the link in the program or welcome kit to join. You will be automatically entered into the waiting room. Our organizing committee will admit you into the hall as soon as possible. If you have questions to ask during the Q&A session, please type your question in the chat box of the Zoom platform and remember to indicate to whom your question is addressed. Ladies and gentlemen, before we begin our conference, I would like to remind all participants to fill up the attendance form in the chat box. Also, I would like to inform that we will be having the first lucky draw session later before the concurrent session begins. This lucky draw is open to all participants. In order to join this lucky draw, you are required to fill up the Google form that is already shared in the chat by 9:30 a.m. today. Just to inform you that the information that you are providing in the form will be shared with our sponsors. And these lucky draw prizes are gift coupons that can only be used in Malaysia. Ladies and gentlemen, with much respect, I would like to invite our organizing chairperson for ICMBB 2023, Assistant Professor Dr. Michelle Su Uy-Yun, to deliver her opening remarks.
Thank you, Yik Ling. A very good morning and welcome to the 6th International Conference on Molecular Biology and Biotechnology 2023, or as we have informally been referring to, the ICMBB 2023. On behalf of the organizing committee, I would like to warmly welcome all of you participants from local and international universities, speakers, and sponsors. I would like to extend my deepest appreciation to our distinguished speakers for accepting the invitation to share their knowledge and experiences with us during this 3-day conference. The theme for this year's conference, Transdisciplinary Research Where Nobody Gets Left Behind, was specifically conceived to reflect the dynamism of research that is moving very steadily towards addressing societal changes. To accommodate a new era where different disciplines are relevant for high-impact research, transdisciplinarity is of great importance to support a long-term balance. It is pertinent that science is geared towards solving problems to ensure the development of effective interventions to meet the needs of the industry, most importantly by involving stakeholders. Science via research should be focused on giving back to the society through applications that improve daily life. Hence, this conference brings together expertise from various fields of science, namely agriculture, aquaculture, environmental sciences, medicine, food, forensic sciences, and nutrition. I hope that ICMBB 2023 will provide an arena for participants to connect with like-minded researchers for the main purpose of discovering and exchanging knowledge, generating new ideas, and establishing collaborations to initiate our theme. This conference has materialized as the product of amazing commitment by the organizing committee, to whom I'm extremely fortunate to work with. A special mention to our sponsors from Collaborative Drug Discovery, CDD, Annalisa Resources, Next Gene Scientific, Aspire Biosciences, and Oval Medic. Thank you for your support. Last and not least, I wish all participants a fulfilling and enriching experience at this conference. Thank you.
Thank you, Dr. Michelle. Ladies and gentlemen, without further ado, please welcome our chairperson for the keynote session for today, Associate Professor Dr. Siti Sarah Othman from the University Putra Malaysia to chair the keynote session.
Assalamualaikum, good morning. Thank you, MC, for the kind introductions. I can see participants are still pouring in very good vibes for today. I'm Sarah. I would like to welcome all to the ICMBB 2023 keynote sessions and technical talk. Today, actually, we have a lineup of renowned speakers and interesting lectures ahead to start with the day one of ICMBB 2023. And I think everyone can hear me well. Please put up any messages in chat in case you couldn't hear anything or the speakers or any other technical issues. I would like to take this opportunity to introduce our renowned speakers for today, Professor Emeritus Dr. John Beardall. He is currently a professor at the School of Biological Sciences, Monash University, Australia, and spearheading the Beardall Research Group. So Prof. Beardall's research group focuses on the physiology of algae in relation to environmental factors such as ocean acidification. And the major interest of the research group is related to understanding the ways in which marine and freshwater microalgae, including the cyanobacteria responsible for toxic blooms in inland and coastal waters, will be influenced by global change. Today, we are very fortunate because he's going to share his insight as our keynote speakers with a lecture entitled Transdisciplinary Research and Paradigm Shift, Some Reflection Based on a Career in Biology. With that, Professor Beardall, the floor is yours.
Thank you very much for that kind welcome. I'm very pleased to have the opportunity to speak to you today about, perhaps as the title says, my reflections on a rather long career in biology. And some of this does reflect on the theme of this conference on transdisciplinary research. So without further delay, I'll share my screen. And up my slides. And there we go. That's good. And I just want to get rid of that. There we are. Okay, so transdisciplinary research and paradigm shifts. I've structured this talk to discuss the early history of transdisciplinary research. I'll then move on to a brief discussion about comparing specialist silos versus large disciplinary collaborative efforts. I'll talk about how I see the impediments to successful transdisciplinary collaborations are. And the other side to that, of course, is to look at what I consider the key to making transdisciplinary research work. I do recognize that there is a distinction between transdisciplinary research and interdisciplinary or multidisciplinary research. And it must be said that most of what I've done in the past comes into that latter category of interdisciplinary. But nonetheless, I think it also does reflect on the transdisciplinary nature of some research. All of these, of course, refer to the involvement of a range of multiple disciplines to varying degrees on a continuum. Transdisciplinary studies, for example, might, in addition to the scientific side of the investigations, also consider things like ethics, legal implications, social effects of research. So there's nothing really new about interdisciplinary research. But when in the past multiple disciplines have been brought together, the result has very often been a paradigm shift in scientific thinking. And I just want to give you a few examples from history as to the sort of things that have happened. Back in the earlier part of the 19th century, Francois Magendie, he was a medic, was interested in what was in the cerebral spinal fluid. He had no idea about how to look at this. So he called upon chemists in Paris to analyze the cerebrospinal fluid. And this is often seen as the beginnings of biochemistry as a discipline, the bringing together of chemists and biologists to discuss things of common interest. Max Delbrück and a team ended up with 3 co-authors, very much conscious collaboration to bring in particular expertise. And this involved a geneticist, a biophysicist, and an atomic physicist. And they applied a very, at the time, novel mixture of experimental genetics and chemistry, and combined this with the then most current theoretical physics, involving things like X-ray technology and what was then contemporary target theory. And they published this paper in 1935, The Nature of Gene Mutation and Gene Structure. And really, this demonstrated the potential of transdisciplinary collaboration to provide detailed physical explanations of biological phenomena. And really, this marked the advent of molecular biology. And of course, we know about Francis Crick and James Watson and the discovery of the structure of DNA. But Delbrück, I think, was probably there before them. More recently, we've seen the emergence of another field that involves bringing in physical, chemical, and biological sciences and converging, and that field's nanotechnology. And I'm sure we can all think of other examples where there's these new sort of incipient technologies bringing together disciplines that had not previously been involved. I want to say a little bit about silos versus transdisciplinary groups. There's a saying in my country, and probably around the world, a jack of all trades is a master of none. And essentially this means is that if you try and, unless you're some real polymath, if you try and look at too many different aspects, you never really master each of them. You're never an expert in each of them. The idea of transdisciplinary or interdisciplinary research is one with an awful lot of traction amongst university management. These days, we're all being asked to consider transdisciplinary research. And as I'll show you, there's certainly a lot of university-driven transdisciplinary research. It's also popular among a number, though not all scientists have embraced transdisciplinary or interdisciplinary studies. They choose to work by themselves. But to tackle complex problems thoroughly does require a deep understanding of disciplinary specific topics. And often, though not always, this involves large teams of researchers who are thinking in the same sort of way, like-minded researchers. So operating in silos of people in the same discipline, you're just bringing together a range of people within the same discipline, is thus often popular. Silos can be very productive. Specialization and compartmentalization in research are, in many cases, critical to excellence. And of course, we all strive for excellence. Most scientists in our training involves being independent. It's part of the criteria for doing a PhD, is to demonstrate your capacity for doing independent research. Merlin Crossley, I put this quote up because I rather liked it, in the conversation recently, talked about the biggest trap for a young scientist is to jump onto the back of tempting new bandwagons. If you just jump onto other people's bandwagons, virtually guarantees that you won't be first to discover anything new. At best, perhaps you'll be second, you'll be recognized for a contribution, but you won't be seen as the leader in a discipline. That said, trans and interdisciplinary interactions can be very powerful and very productive. And throughout my career, I've been privileged to collaborate with people from a range of other disciplines. And I like to think this has broadened my outlook and actually improved my ability to appreciate the way algae, that's my research specialization, function in the environment. And I want to give some examples of this as we go forward. All of the environmentally focused multidisciplinary projects I've been involved in have allowed me to work with experts in their own disciplines. For example, the physical and chemical hydrology, people working in geochemistry, and this has allowed us to interpret in a much more holistic way the biology of the algae that I'm interested in terms of changes in their environment, in terms of interactions with other organisms such as marine invertebrates and fish and so on. And these sorts of collaborations provide much deeper perspectives into a problem than we, as a single lab, would not have been able to provide. Climate change is, of course, a major problem at the moment. It's a great example of a big problem in need of a transdisciplinary approach. Assessing the effects of climate change, possible mechanisms for amelioration of these effects involves inputs from a huge range of disciplines. You only have to look at the IPCC reports to get a feeling for how broad that coverage is. Look at those reports, you see materials from biology, from chemistry, physics, meteorology, social science, health science, and the list goes on and on and on. And that's a really good example of the power of transdisciplinary research applied to a major problem that we face in the future. I want to talk a little bit about some of the examples from my own work. I've worked with engineers, for example, a fair bit, to devise, for example, new ways of generating power. And the first paper up here on the top right, I was involved with Professor Fang-Xiu Moy and her colleagues, driven through University of Malaya, looking at algae in photovoltaic devices to generate bioelectricity. I've been involved with engineers at Monash to devise new ways of dealing with blue-green algal blooms. Can we use electrochemical inactivation of, for example, microcystis, a very problematic blue-green alga, to remove not only the algae, but also to destroy the toxins, the microcystins. And indeed, things we came up with was to develop a floating system that you could put onto a water body, float around and destroy the cyanobacteria in situ. Perhaps the most challenging disciplinary project I've been involved in was the very large study looking into the environmental health of Port Phillip Bay in the early 1990s. This was a government-funded project, but it was huge. It involved a total of 47 research tasks, ranging from just a basic background scientific literature reviews to a host of full-scale field surveys, including studies in physical oceanography, toxicants, both biological and chemical, algal nutrients, marine ecology, and modeling. And my group were involved in quite a few of these tasks. Needless to say, with a team that big, 47 individual projects, requires a lot of very careful management. And CSIRO, who managed the project, set up regular discussion groups very early in the planning of individual tasks and also throughout the study. And this was the key to success of this major project, because we had access to information on most of the other tasks that were being carried out. And this allowed us to better refine and design our tasks and modify them if necessary, and to interpret our data in a better way. The other form of trans or interdisciplinary research I've been involved in centered on collaborating with people who bring entirely new levels of approach and instrumentation to the party. And again, this enabled new perspectives on biological systems that would not otherwise be evident. For instance, one of my postdocs, a fellow called Phil Harrow, was friends with a chemist at Monash, Baden Wood. And Baden was working on infrared spectroscopy. And Phil realized that this was a tool that could be used to follow changes in macromolecular composition of algae under environmental changes, such as global climate change. And this developed into a major field of study, which eventually led to further interactions with other disciplines, synchrotron scientists, the physicists and the modelers at, for example, the Australian synchrotron, the Bessie 2 synchrotron in Berlin, and the Advanced Light Source at Berkeley. And that allowed us to probe aspects of algal biology at the single cell level, and even do imaging of macromolecular changes in live single cells. And this came, just a few of the papers we published on this. Our very first paper in this area was to look at infrared microspectroscopy to discriminate between cyanobacterial strains as a quick and relatively cheaper way of identifying cyanobacteria compared to, say, DNA sequencing. We looked at how infrared spectroscopy can be used as a nutrient concentrations or CO2 levels. And we even did mapping of nutrient-induced biochemical changes in living single cells using synchrotron infrared spectroscopy, where you can get a very high resolution. I'll just show you an example of that. Here's a living cell of an alga called Microsterias. It's a desmid. We did cheat a little bit because this is a rather large cell. It's perhaps a couple of hundred microns across. You can almost see it with the naked eye. But if you do infrared spectroscopy and focus on the protein, you can see this is the living cell, this is the protein image, and you can see the protein is concentrated in the center of the cell where the chloroplast and all those vital functions are occurring, and not very much protein out in these arms that you see in the ordinary micrograph here. And you can do time sequences. We could put a single cell into a chamber and then examine that over a period of time, about 100, in this case, I think it went up to 24 hours or thereabouts, sampling regularly. And as you go up here, starts off, this is a phosphate limited cell that has lots of stored lipid. And as you resupply phosphate over time, you can see it goes from these darker blue colors and green colors through to the lower levels of phosphate represented by the yellow color. And you can do this to any point on the cell. This just happens to be a transect measured across here. So you can probe what's happening in an individual cell at any point in the individual cell over time in a live cell. And that was pretty cool. It was something we wouldn't otherwise have been able to do. The other thing we've been looking at are things called coccoliths. These are calcium carbonate scales formed in vesicles of these organisms, such as Coccolithus pelagicus. Previously, you could only see these under electron microscopes. And we wondered whether we could visualize coccolith formation better using soft X-ray tomography, which allowed us to go and play with big toys, like the Advanced Light Source at Berkeley. Here it is. Here's the Berkeley campus surrounding it. And it's this massive building with a heck of a lot of very high-powered instrumentation. And these are our collaborators from the ALS. The synchrotron, of course, allows you to pull off beams of energy, X-rays, and infrared beams as well at any point. So all of these represent sites where you can set up. We worked in the National Center for X-ray Tomography down here, and it's well set up with a whole heap of really interesting gear, but unless you're a physicist, I wouldn't have a clue how to work any of this. So working with a physicist was really, really important. This is the beam coming in from the synchrotron through here. And then you have this, you put your sample in here and look at it doing X-ray tomography. And you get sort of images like this, the series of images through the cell, and then involving the modelers, the people who are expert in this, you can then build up a image of what the cells look like. These are just more tomographic images. So you can build up an image of what a coccolithophore likes. In this case, we focused on the outside with these liths. This is another image taken from the Bessie 2 synchrotron. You can start to see fine structure of the coccoliths, and you can build up pictures of cells in this way. And this is really cool stuff that you couldn't really do unless you involve synchrotron science and physicists. I want to talk a little bit about some of the impediments to making this sort of thing work. Large groups can be prone to difficulties in acknowledging the roles of individuals. People get lost in very large groups, and that is a problem. You need to make everyone feel valued and their contribution valued. There are, of course, differences in approach, differences in terminology, and differences in expectations of what you're going to get out. So, for example, the synchrotron physicists might have a different terminology. They probably have different expectations of what we were hoping to get out of the research on Bessie or whatever. I found there are particularly different expectations in industry. Industry has a need for rapid solutions. Some of my experiences have been good. The industry partner, particularly if they come from a research background, is more likely to understand what it's all about. Others are less easy to run. I've been in situations where the industry partner doesn't really understand the scientific process and expects almost instant returns on their investment in research. So you have to be a little bit careful about this. What can you do to make it work at an institutional level? Well, some institutions have actually been deliberately established to foster transdisciplinary collaborations. I said at the beginning, a lot of university management these days is very keen on transdisciplinary or interdisciplinary research. Just some examples. The Federal University of Minas Gerais in Brazil has the Institute of Advanced Transdisciplinary Studies. Northern Illinois University has a Center for Innovative Teaching and Learning, bringing in different approaches, transdisciplinary approaches to developing learning techniques and so on. And Indiana University have the Environmental Resilience Institute set up specifically to look at transdisciplinary approaches to environmental problems. One really good way of doing this is to, rather than just get away from having silos, but just simply build bridges between the silos. So you've still got that concentrated expertise within the silo, but you link different silos. Rather than just break it down, you just bring together different expertise in different silos. Shared infrastructure is a really useful way of bringing people together. It's a very efficient way of linking people into teams. It's efficient in terms of use of the infrastructure because if it's done properly, it can run around the clock. It needs to be maintained by dedicated technical staff and upgraded as required. And that leads to another problem I'll talk about in a second. But it's a really good way of bringing people together. In Australia, we have the Australian Research Council funding what are called Linkage Infrastructure Equipment Facility, or LEAF grants. These and another set of grants, the National Collaborative Research Infrastructure Strategy, or NCRIS, are really important by providing funding for shared instrumentation and shared infrastructure. you can target interdisciplinary research. Monash had its own internal interdisciplinary research grants to foster research that leverages the breadth of Monash's expertise, bringing together disciplines to tackle major research challenges. And these comprised either major project grants between $1 and 250,000 Australian dollars, or just seed grants, which are somewhat smaller, 25 to 50,000 Australian dollars. And I had one of these working with an engineer and an architect. The architect came along with an idea of building windows that were hollow, and you could grow algal cultures in the hollow, like in the double glazing, between the double glazing, if you like. And this would provide a way of stripping CO2 from the room, releasing more oxygen into the room and making the environment, apart from also looking nice, a little bit more easy to cope with. And other universities do the same. I just wanted to say the important thing down at the bottom there is the sustainability of funding is important. You need to ensure that these schemes are sustainable in the long term. Faculty of Arts at Monash runs undergraduate units in interdisciplinary research, bringing together a whole lot of different people. So right from the beginning of education, you're making people think about interdisciplinary research. University of New England in Australia, again, offers a Bachelor of Sustainability, combining subjects from arts, business, economics, science, geoscience, whole heap of different disciplines. And they graduate as specialists in sustainability, but with this broad transdisciplinary background. Some journals promote trans or interdisciplinary research. The Royal Society has this Journal of the Royal Society Interface. Again, it designed specifically for people to publish work that has been at the interface between life sciences and physical sciences. The key to making it work at a personal level. Well, I've been lucky that projects I've been working on went smoothly because of a number of things, as I see it. Each of us knew the other partners very well. Even dealing with a lot of the corporate bodies, we knew the people involved. In fact, several had been Monash graduates. At the individual level, each of the main universities or university contributors knew one another. We talked regularly. We had a healthy respect for each other's work. In many cases, we actually shared supervision of PhD students. So we were building on pre-existing interactions between our labs. And of course, the more you work together, the more this stimulates new ideas. Importantly, you need careful planning and discussion of individual roles in the collaboration at a very early stage. It really helps. It's important to have a respect for collaborators outside your own discipline. You must be willing to listen to their take on the problems being investigated. Be open, be willing, and be flexible. Be willing to be flexible in your approach. When you're talking to someone, they come up with an idea, just don't dismiss it. Think hard about it, and perhaps modify your thinking a little bit so you can incorporate that into the into your project. So talking a lot, bounce ideas around an awful lot, and other ideas, other approaches appear. Wouldn't it be good if we brought in this person with another set of expertise? We can bring that in to this transdisciplinary project. And if you do all of that, that, to my mind, is a key to success. Some useful references are here to talk a little bit about transdisciplinary research, why you might want to do it, analysis of how does it actually work, and right at the end is the science in silos isn't such a bad thing. I'm running out of time, so I better thank you very much for listening to me, and I wish you well in your future transdisciplinary endeavors. Thank you.
Thank you so much, Prof. It's really such an insightful. It's like hearing back. I remember my mom always tell me to be jack of all trades is master of none. So that is a very strong saying, especially for us in transdisciplinary approaches. And I like the way you approach it, how we are actually solving the world problem, something like as sustainable development goals. And in Malaysia, we also encourage to work scientists, especially through transdisciplinary approaches. And we have come up with this 10-10 My SCIE to address socioeconomics based on the strength of science and technology in the country. Thank you again for such an inspiring talk and a lot of advices for all of us here. There is no Q&A for Prof. Bearder, but we will actually forward the questions later to you.
That's fine. I'm happy to deal with any questions offline. That's terrific.
Thank you so much, Prof. We will share your details later to our participants as well.
Thank you for the opportunity to speak to the conference.
Right. So moving forward, we have another technical talk, another renowned speakers from the collaborative drug discovery, Dr. Eric Gifford. He's the business development scientist. Dr. Gifford is an experienced pharmaceutical R&D and IT professional with in-depth computer-assisted drug discovery, development, and pharmacovigilance knowledge. His scientific research was primarily focused on computational models useful for understanding drugs, biological targets, and physiological response relationship using a variety of machine learning and AI methods. It's very hot topics these days with generative AI especially. Prior to joining CDD, he was Director of Research and Development Informatics at the Singapore branch of MSD Pharmaceuticals. We are very honored to have Dr. Gifford today with us to share his insight entitled CDD Vault, a research informatics platform for enabling biotech collaborations. Hi, Dr. Gifford, you there?
Hey there, thank you for having me. I think I'm going to have some problems sharing the screen. So I'm getting a message here, it says host disabled participant screen sharing.
Right.
So I don't know if that can be fixed on the other side or if someone wants to.
Okay, the host will allow you to share your screens, or are we sharing Dr. Gifford's screen?
I was trying to hit the share screen button.
Yeah.
And it comes up. Yeah,
okay.
No problem. I can give a brief introduction to myself and the company. So what I'd like to do today, first of all, is thank you very much for the invitation and say that Collaborative Drug Discovery is pleased to be a sponsor of this conference, the 6th International Conference on Molecular Biology and Technology and Biotechnology. I'd like to express our gratitude to the organizing committee for making this event possible. My name is Eric Gifford, and I'm responsible for business development for CDD in Southeast Asia. I have many years experience in R&D with Big Pharma, most recently retiring from MSD Singapore, where I was the research and development informatics site head. Today, we'll have a brief introduction to CDD, and then as time permits, look more carefully at how CDD is supporting collaboration between scientists by hosting public data sets and a freely available visualization tool that we will show a demonstration of a bit later. Here we go. Let me push that over here. Apologies for the technical.
I think you can share it, your screen now.
I can.
Perfect.
Let me do that now.
We're waiting for it. Okay, we can see your screen. Thank you very much, Dr. Gifford.
Okay, let me go full screen mode. Swap out the displays. How does this look? Do you see my full screen?
We are seeing the presenter slide. Maybe you have to swap display.
Swap displays.
Yeah,
swap
perfect.
display.
It's perfect now, yeah.
And let me proceed here. You can see now? All right. Collaborative Drug Discovery is a global provider of software used to help accelerate drug discovery by effectively managing your research data. It allows scientists to focus on science and not data management. CDD was founded in 2004 by Dr. Barry Bunin, who observed at the time that the work of researchers was severely obstructed by difficulties in sharing and reusing data securely. Some used paper lab notebooks, others used early versions of electronic notebooks, and still others shared data on Excel spreadsheets. In all these cases, it was difficult for research teams to work together and share a single source of truth about their research data. With this as the problem to solve, Barry went on to found CDD and his team continues to develop CDD Vault with continuous improvements and enhancements. CDD Vault is now used by thousands of researchers worldwide, including those who work at universities, research institutes, biotechs, pharma companies, CROs, and more. So let's talk a little bit about what is CDD Vault. Well, CDD Vault, Collaborative Drug Discovery, the provider of CDD Vault, provides a comprehensive platform for managing molecules and biologics alongside data that you collect and analyze to support your research. Imagine how you are managing your research today. Perhaps it is in paper notebooks or spreadsheets and documents. What CDD provides is a simple to use, single, secure electronic platform for your discovery and preclinical research data. CDD Vault is made up of the 4 modules that you see here: activity and registration, inventory, visualization, and an electronic laboratory notebook, or an ELN. Each of these modules is designed to provide a user-centric experience for scientists whenever or wherever they are. The electronic lab notebook is used by scientists in much the same way pen and paper notebooks have been used for generations. However, the electronic notebook brings capabilities far beyond the traditional laboratory notebook by seamlessly integrating all of the other CDD modules. Compounds can be registered, experiments or chemical synthesis planned, and results can be captured and published using the ELN. Activity and registration is the module where chemical compounds, biologics, and associated data can be added or uploaded to the CDD Vault database. It also is accessible and searchable through an intuitive, user-friendly, web-based interface. We will see in the next slide specific examples of the types of data that can be stored in CDD Vault. Inventory is the module where scientists can track the location and amounts of chemicals, biologics, and reagents in their labs. It is user configurable and adaptable to fit your specific workflows and processes. And the 4th module, visualization, allows users to visually analyze and mine their data for interesting and useful relationships. There's also a no-cost version of this module available to scientists, which we will discuss toward the end of this presentation. As I previously mentioned, CDD Vault is a very flexible scientific informatics platform and personal secure database that is highly configurable by scientists to accommodate a wide variety of data types. The slide shows some examples of the different types of data users typically store in CDD Vault. As you can see, the data types range from small molecules to biologics, chemical synthesis, mixtures of chemicals, in vitro and in vivo data, as well as imaging and analytical data. All this information is searchable from the CDD Vault web-based graphical user interface. And although this slide refers to the IT architecture of the platform, it's important to note that you don't have to be an IT expert or have a dedicated IT team to set up and use this platform. Typically, these platforms are set up by customers with the help of CDD support staff and ready to use after about a 90-minute training session. It's a very simple system to deploy. So what I'd like to do now is bring your attention to the commitment of collaborative drug discovery to open innovation and, as the name implies, collaborative research. CDD provides no-cost access to a set of highly curated data sets and a no-cost visualization tool, each of these accessible at the URLs shown on this slide. There are currently more than 140 public data sets with more than 2.9 million registered compounds, and a wide range of endpoints specific to each data set available to users at no cost. I encourage you to explore those data sets. Another offering is the visualization tool. We'll spend the remainder of our short time together talking about the CDD visualization tool. What is provided in the CDD visualization tool is the ability to allow scientists to upload their data and plot, graph, analyze that data in a wide variety of ways. The data never leaves your computer, and the application runs in the browser window on your computer so that your data stays securely with you. Go back one here. Okay, so let's walk through an example use case to highlight the capabilities of visualization. While at the same time, think about the question, how do you manage your own research data? In my long experience collaborating with academic, institute, and industry scientific researchers, the tool of choice for recording data and sharing data, assuming we have moved beyond pen and paper, is Excel or similar spreadsheets and documents. So I'll focus on that, that format as our entry point into the CDD visualization tool. So what I show on the screen here is a typical spreadsheet with data. And this is formatted in the correct format for uploading into the visualization tool. So it's good to have the rows as observations. For example, in this case, we have set a set of anti-inflammatory drugs. The chemical structures are represented by smile strings, and the other columns are experimental or calculated results associated with that compound. For example, molecular weight, log P, number of hydrogen bond donors, number of hydrogen bond acceptors. And off the screen, if we could scroll over to the right, you'd see the KIs of the compounds screened against cyclooxygenase 1 and cyclooxygenase 2 assays. And we'll see this data as we proceed. So the first page you see when you go to the visualization tool looks like this. What you do is you click to upload, and the visualization tool will automatically detect and interpret the columns in your spreadsheet. You also see here that we have some example data sets if you would like to explore, but don't have your own data. Okay, once that data is uploaded, the first thing you'll notice is that the user is presented with a data table similar to the one you see here. Most obvious is that the chemical structures were previously represented as smile strings have been converted to chemical structure diagrams. Double-clicking on a structure brings up a chemical structure editor useful for selecting substructures and searching for those substructures in the underlying data set. On the right side of the screen, you see bar graphs showing the distributions of the various properties that you imported. Sliders on the bar graphs can be used to select data based on the individual properties. In addition to the table, Visualizer has a variety of plotting capabilities. For example, we have a 2D scatter plot. In this case, we show log P versus molecular weight. It's a very interactive plot, and the markers are colored by one of the experimental endpoints, COX-2 Ki, and sized by COX-1 AI. So you have about 4 dimensions all on the screen here at the same time. We can see from the other icons at the top of the visualization that we can also plot 3D scatter plots, histograms, bar charts. In addition, more chemical structure analysis capabilities are available, such as substructure searching, R group decomposition, and clustering based on chemical structures. The next slide shows an example of this chemical structure awareness. Not only can we plot the experimental data, we can also highlight the chemical structures on the plot. Putting the mouse over a point pops up an image of that chemical structure associated with the point. In addition, I've selected 3 compounds and pinned the structure and associated data to the points, as you see in these cards. These summaries or cards can be interactively moved anywhere on the plot while still retaining the connection to the plot marker. This capability is also available in the 3D scatter plot. Finally, I would comment that any of the plots can be exported as publication quality graphics in either PDF or PNG format, or the Excel file can be exported with the additional features such as the calculated properties or the cluster memberships that were added during the visualization process. Unfortunately, I did not have time to give a live demo or to cover all of the features, but I hope that this quick tour has caught your attention and motivates you to explore either on your own or with my assistance. I encourage you to contact me directly for more information. What I'd like to end with is I'd like you to think about this and remind you that CDD Vault brings value to scientists all over the world by allowing them to focus on what they do best, great science, and not necessarily data management. Thank you. And this ends my presentation. And please do reach out to me if you'd like a personalized demo, or if you'd like to just run through or you have some questions about the visualization tool. Always there, happy to help at that. That's my direct email, egifford@collaborativedrug.com. Or for more information, including a lot of resources, including a webinar series that Collaborative Drug Discovery puts on, and recorded versions of those webinars, all available at that website, collaborativedrug.com. Thank you.
Thank you so much, Dr. Gifford. Right. So there's a contact numbers, there's contact emails for everyone to reach out directly to Dr. Eric Gifford. So it's a very informative talk, and I would like to now open the Zoom hall for questions. You can either unmute your mic or write in the chat box. Maybe I'll start the ball rolling. When you talk about the CDD vault and about data management, it's some things that, well, scientists really keen to, because managing information is not really something easy. I'm intrigued to ask further about your interest in open science, because it's taking an effort for scientists to actually share some things that they discovered that they think they would have protection towards it. Recently, Malaysia, we just launched it's our own Malaysia Open Science Platform, MOSP, throughout the countries, and we have networks through the universities. But I still felt that researchers are still not keen or maybe even convinced to start or continue sharings. So what is your thought on this, and how to further encourage researchers to confidently share and support open science platform?
Yes,
Maybe you can share your insight.
No, it's a good question, and it's one that I've struggled with, had problems with early in my career, but as I get later and older in my career, I think more about the good of society and less about the individual good of the researchers, right? So you have to understand, I work for a big pharmaceutical company, and in big pharma and biotech and in universities, more often than not, IP is your lifeblood, right? Your intellectual property. And so how do you protect your intellectual property when you're entering into an open innovation or open collaboration? And that's a really difficult question to answer, and I think there's a range of answers to that, right? I admire and would like to talk more about Malaysia's efforts in this, and my colleague Mark Garlinghouse is on the call in the conference as well. We'd both like to talk to you, because we are spinning up some initiatives around open science, open data management, and so on. What I can say, back to collaborative drug discovery and the vault, is that you don't have to share everything, right? So I hope I didn't leave the impression that if you have a vault subscription and you enter that is your own personal vault, and you have the ability to assign or remove privileges for individuals at the individual level into the various projects, right? So you can completely open it up to all members of a project or to all the individuals, or you can restrict access in a very granular way. I wish I had a really easy answer to a very hard question. No, I know. But if we could figure that out, it's all about funding, incentives, and global good at some point.
And it's also perhaps a policy to also highlight if there's a misconduct or misuse of data from the open science platform. I think we need more regulations, just like how generative AI is coming up, and there's no clear regulations perhaps for exploitation of those tools. I think it goes line in line, and like you said, it's not something that you can answer in one line. But I think a lot of our audience would have those burning questions in their mind as well. Right. So I'm just looking at chat now. Browsing through, so I think everyone's still warming up since day one, but you got Dr. Gifford's email. I'm sure a lot of you have screenshot those slides as well, also with our Prof. Beardell just now. Thank you so much, Dr. Gifford, for your sharing information. And please share with us the MSMBB's, the upcoming webinars from CDD that we can share with our members to also watch. Thank you, Dr. Gifford.
Very good. And I'll just put my email in the chat window as well, in case you didn't jot it down.
Yeah, right. Thank you for that as well. Yeah. So before I pass the mic to the MC, I would like to remind every participant, every one of us here, to fill in the Google Doc for your attendance, because 50 ringgit Grab vouchers are waiting for us at the lucky draw. All right, thank you again. Thank you for being with me and two speakers, Prof. Beardell and Dr. Gifford, through the keynote and also technical talk. Back to you, Prof. Dr. Chew.
Thank you, Associate Professor Dr. Siti Sarah Ohman, for chairing the first talk session for today. Ladies and gentlemen, thank you for still being with us. Now, we will have our lucky draw session for today, and we will select 3 lucky draw winners. Here's how our lucky draw works. Our committee will spin the wheel, and the lucky draw winner's name will show on the screen. Good luck, everyone. All right, here we go. Our first lucky draw winner is Hamiza Shahira binti Hamiza. Congratulations. Okay, our next lucky draw winner is Hasliza Hassan. Congratulations. And our final lucky draw winner of today is Isrin Izati binti Razali. Congratulations to all our 3 lucky draw winners for today. Ladies and gentlemen, we will have our group photo session for today before we proceed to the concurrent session. This group photo will serve as a keepsake, reminding us of this special occasion. Your smile will make it an even more cherished memory for all of us. We understand that personal circumstances may differ, and we respect everyone's choices. We kindly invite you to join us with a smile, and I appreciate it if everyone can turn on your webcam of your device if you are comfortable and able. Our organizing committee will capture the group photo. Okay, everyone ready? Okay, 3, 2, 1, smile. One more snapshot. 3, 2, 1, smile. One more. 3, 2, 1, smile. And the last one. 3, 2, 1, smile. Okay, thank you, everyone. Okay, thank you, everyone, for creating a picture-perfect moment together. And next, we will have the concurrent session for today at 10 a.m. later. And we have arranged two concurrent sessions for today. One session is on the agriculture, aquaculture biotechnology, and molecular biotechnology. We will also have another session that is related to medical, forensic biotechnology, and molecular biology. You may join the respective hall for the concurrent session in which you are interested. Before we proceed to the respective hall, I wish everyone a great day ahead. Thank you.
Hi, good morning, everyone. So welcome to Hall 1. I'm Boon Jin from So I will be serving as the chair for these sessions. So I would like to inform you that these sessions focuses on agricultural biotechnologies and molecular biology. So if you are interested in attending the medicals or forensics biotechnology and molecular biology sessions in Hall 2, so please refer to your program group for the link. So today we have an exciting lineup of speakers. So we will begin with plenary sessions featuring one distinguished speaker, followed by two invited speakers who will be sharing their expertise in the field. So let's get started. So we are honored to have Professor Rofina Yasmin as our plenary speaker. So Prof. Yasmin is an honorary professor at the Institute of Advanced Studies, So she is also an adjunct professor at IUMW and an honorary fellow of the Tun Ghazali Safi Institute for Strategic Leadership at UCYP. So besides, Professor Yasmin is also a fellow of the Academy of Sciences Malaysia and is listed as in the top research scientist Malaysia database. So she is still very active working on her research, focusing on utilizing modern biotechnology strategies to tackle fundamental problems in agriculture, as well as issues related to biopolicy, bioethics, and biosafety. So Prof, you have 30 minutes and I will let you know 2 minutes before the end. So, and to all participants, kindly post your questions in the chat box. So without further ado, let's welcome Professor Rofina Yasmin. Prof, the floor is yours.
Thank you very much, Boon Ching. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all. And firstly, I'd love to thank MSMBB, society close to my heart, for the invitation to share and speak today. So I'll share screen right now. I think, is that it? Is that okay?
Yeah, we can see your slides and hear you.
So I was very honored to hear this morning's talks, and they really set up a kind of a launch pad for what I'm going to share today with all of you. And it is the concept that we have to embrace transdisciplinary research or transdisciplinary approach, especially when we are trying to really bring our technology out into application. All the research that we've done has to come to a conclusion that it can benefit, the end user, whether it's the public or farmers in the case of what I'm going to talk about today. So my title is to accelerate the adoption of molecular technologies for plant health and crop biosecurity through a transdisciplinary approach. Where's the button? Okay, we know this fact, and I think scientists, as we do our research, we have to be really aware that out there the world is facing catastrophic problems. The population growth is really going to exceed 10 billion people by 2050, and we need to feed all these people. But there is, a perceived shortage that is expected if we don't do something drastic about how we manage our planet and how we manage our resources. And so what are the approaches that we need to do to really increase or really meet the demands of food? Of course, we can just have more land use for agriculture. And you have heard that we really need to decrease food waste. But what people don't actually realize is how much food or potential food we lose to pest and disease. And I'm must say that COVID-19, as much as it was painful for the whole world, really brought into highlight how important it is for us to keep ahead of pathogens and pests that may become a big threat to our society, be it for health or in this case for agriculture. So much so that the United Nations, and I'm not sure even many plant scientists know this, that the 12th of May every year has been designated the International Day of Plant Health. And this really is to raise the global awareness of how important plant health is to reducing hunger, to reducing poverty, to protecting biodiversity and environment. So it is about planetary health. To protecting the plants will protect life. So that's the slogan. And it's no small matter, because if you are into climate change, we know that plants are essential for carbon capture. If you are an environmentalist, we know that plants are important for remediation. And if you are an agriculturist, you know how important it is that plants feed both humans and animals. So what is the impact of plant health to the planet, people, and profits? Because as we say in transdisciplinary thought, it's not just about applications to serve the public, but it is about continuity. So it is about sustainability. So on global average, 10 to at least 30% of crop production is lost to pests. That's a huge amount. And we're not talking about post-harvest losses, food waste. We're not talking about pest contaminants that make food inedible. Just in the field, 10 to 30%, up to 50%. And history has shown us that destruction of farmland due to pathogens has created changes in world history. The potato famine as far back in history, and even more recently, when coffee has been affected by coffee rust. And I don't know whether you heard the news yesterday. If you are a fan of Robusta, you're in trouble because the crop has suffered great damage and the prices of coffee and espresso is going up. But more importantly, more than in the last pandemic of coffee rust in South America, more than 50% of people in the industry there lost their livelihoods. More than 400,000 people, almost 500,000 people lost their livelihoods because of disease. So it affects availability, access, utilization, and stability. And therefore, it introduces the term to us today of biosecurity threat. So we know now what biosecurity, now everybody knows what a biosecurity threat is. COVID was a biosecurity threat. It threatened every aspect of our life, not just our health, our economics, our ability to travel, our access to everything physical. And in agriculture, it is the same. Emerging plant diseases have the potential to be a catastrophic biosecurity threat. And emerging plant diseases include any disease that have increased in incidence, geographical or host range, any that have changed their pathogenesis. At one time, they could be just a benign organism, but due to changes in the environment, they may have become pathogenic, newly evolved, or newly discovered. And there are literally millions of microorganisms that have yet to be discovered and recognized. And impact of climate change, we cannot run away from that when we talk about pests and pathogens of agriculture. So recently, the FAO, last year, in fact, it was last year, 2020, 2021 maybe, came out with a scientific review of the impact of climate change on plant pests. And this review is really a wake-up call to all of us working on plant pathology. It puts forward that there is a global challenge that we need to face to prevent and mitigate plant pest risk in not just agriculture, but forestry and in the ecosystem in general. And if you look at the heat map down there, you know why. So this is the predicted heat map of how the world is going to be in 21. I don't even know how to say that, 2100. So data on managed systems shows serious gaps in information, however. If we're talking about mitigating biosecurity threats by pathogens, we have to at least know what that pathogen is going to be, or even what that pathogen is that pose such a threat. And what it is now with plant pathology is there is huge gaps in information. Most of the information and most of the funding has been focused on economic crops in limited regions. And there's nothing wrong with that. When you have large scale growing of particular crops, you are definitely more susceptible to pandemics. You're definitely more susceptible to biosecurity threats, and hence a lot more funds have been focused on protecting such crops. Even in Malaysia, in terms of our plantation crops, there has been a lot of good effort in making sure that we are on top of the pests and diseases. But there are really hardly any information, very poorly curated information on indigenous and unmanaged systems, indigenous crops, small crops, and even my favorite crop, banana. Okay. All the focus has been on plantation banana rather than on the diverse banana that's grown by different local communities around the world. So what kind of data is that? So here we are in a molecular biology conference, and of course we are interested in genomics data. And in effect, genomics data of pathogens really can give us a wealth of information that can transcend the lab and ideally move all the way into the field to provide solutions and strategies to farmers, agriculturists, plantation owners, to really manage any threat to biosecurity that may come about. So it is really moving molecular information data out to application. So the essential information will provide means for us to develop rapid detection procedures, and I'll talk a little about this. And very importantly, and I'm glad the speaker before me really set the scene of how important analytical tools are in understanding the data that we have. So predictive analytical tools, and I hope the Vault will move also into agriculture sector soon. Because the scope of work and the threat of to planetary health is just as huge in terms of our food production as it is in terms of our healthcare system. And gene modification strategies, what are the issues there about the use of gene modification strategies? So if you talk about plant pathogen genomes, you can just look at the NCBI database, and there's a huge number of genes and whole genomes of plant pathogens ranging from virus and bacteria and fungi. And this has been growing exponentially because we all know now that the cost of sequencing is phenomenally different from what it was 20, 30 years ago. You know, when I was doing my PhD, now should I say how long ago? Almost 40 years ago, I guess. You know, it took me 2 years to sequence and analyze a 10 kilo base virus genome. So, you know, but we did it, you know, step by step, A by A, T by T. But things have changed phenomenally since then. You know, you could do a genome like that in half a day. You could do the analytics in the same day. You know, it's just opened up enormous potential. But is it really there for us to use? That's my question. So if we look at the status of plant pathogens, sorry, it's a typo, the genotype and phenotype databases, there is really very few curated pathogen database across the plant kingdom. So what do I mean by curated? Of course, if you go into NCBI, you can pull out, say, all viruses that, you know, have related to banana, then you have to start curating it. But there are very few that is dedicatedly curated, except for a few crops that of economic importance that do curate some databases of that are dedicated to that particular crop in particular regions. And many databases that have been started in the past and have not been sustained. And for us in Southeast Asia, there is no real accessible resource, that tracks what is really going on around the region. Some countries might be focusing on certain pathogens for a certain amount of time, and other countries differently. But pathogens doesn't know borders. Vectors do not know borders. And as a region in Southeast Asia where we share similar climates, similar indigenous crops, really it is a state that is quite alarming. And we as molecular biologists should maybe get together and address this access to information, because people do work on genomic or gene data of indigenous pathogens, but where is the data? We really have to spend a lot of time curating this, and even when they have been curated in the past, they have not been made public. So we need to put these genomes to work, really. We need to put these genomes to work. I mean, this review actually just highlights what you could potentially do with all these genomes. They looked at how many plant pathogenic bacteria genomes there are in the database, and it is phenomenally huge. And the review kind of said doesn't really show how they've been used, but rather the potential of how they could be used, and really the opportunity to be able to now access this database, to be able to do the analytics on it, really probably is in the best time. So unlike a lot of other genomes, maybe in the medical field, most plant pathogenic organism genomes are really open access. And one of the reasons is because the commercial potential has not really reached a stage where it excites investors or commercial interest. And in that sense, it gives us a window of opportunity to really be able to exploit this data for studies on whether you are looking at fundamental studies, understanding molecular mechanisms of plant pathogen interactions, whether you want to try and understand why some plants are more susceptible than others, whether you want to just identify accurately what are your causal agents. Is it the same causal agent in my country as it is across the border? Taxonomy has always been a problem with microorganisms in plants, understanding population genetics. And at the end of the day, can we use this information to really create some approaches or strategies for control of these pathogens? So huge opportunity. Now, I got very excited when doing this talk because I didn't realize that a website or a database, which I was very sad to find was closed in 2013 due to lack of funds, because it was run by a group of researchers who just ran out of funds that they could put together to run this database, has been taken up by the AAB. And this is the plant virus database, or dpvweb.net. So it stopped in 2013 and it just got revived recently, and very small number of sequences in it compared to what is in NCBI, because they are depending now on really members of the society and also virologists to really input their whatever data they have and curate it properly into this database. So very exciting. I was very excited to see that they did incorporate on the right there the virus I did for my PhD a long time ago. And as I said, a tiny virus which really took half my PhD to simply sequence. But it was a milestone of sorts for me in terms of getting excited about what the technology could offer. And right now, really, my wildest dreams have been realize, with the kind of analytics that you can do, the speed of sequencing. And I wish that I was 30 years earlier in my career. So all those young scientists out there, you are in such a good place where technology is concerned and where transdisciplinary research, as the speaker before this said, you don't have to be a computer scientist now to do deep tech analytics. You don't have to. You have to have just access, and you have to have collaboration. And again, Prof in the first keynote stressed that nothing works nowadays without collaborative research. If you really are serious about translating your work to good application, you need to have a really transdisciplinary team on board. And what do we hope to get from this kind of database? So I just brought in the word that I really started my career with, which is looking at banana viruses. I was really disappointed when I came home to find that we are the center of biodiversity of bananas, but literally we had no idea what kind of pests or pathogens other than the major ones was present in our environment, was affecting our indigenous crops. And so we did try to initially look, script funding together to do simple surveys. So, I did a survey on a virus called banana streak virus in 1999, and then again in 2001. And the diversity of this just one virus, even in the low levels that we detected it in Malaysia, was quite astounding. And on the right, really, is a very recent review of banana streak virus sequences in the NCBI database. And it just shows that the genome is so diverse around the world, and that, there is a need to pay attention to viruses and diseases like this, as future threats to the banana economy and even to biodiversity. And, the problem was at the time was really, how do you do large-scale surveillance of these plant pathogens, the costs, the low throughput. But, people keep working on it. And now even for banana viruses, a recent paper last year by Taiwanese researchers came up with an improvement, a big improvement to the multiplex that we've been using. They use something called liquid chip, which is a bead assay PCR-based test. And they could do up to 100 assays in one go, multiple viruses in a single tube, and really fast analytics. And really, this is the kind of research that we need to really enable us to start a proper surveillance program, to enable us to make some plans, at least, to mitigate threats of pathogens. Right now, the way to mitigate it is, if you have a disease crop, you just cut it down and burn it, and then hope for the best, that the pathogen dies with it. But most of the time, we do not actually know what is the pathogen that is circulating. And if you look at why so little work has been done on virology in banana growing countries, is because there is overriding a fungal disease that really took the world by storm, and it's still a pandemic around the world right now. And first discovered in 1967 in Taiwan, and then it spread in Asia, and it spread and spread and spread. In 2016, the FAO really regarded it as an endemic pathogen already, beyond pandemic, and that it had spread by 2021 even to Latin America, which is the largest banana growing area in the world, and that really has put fear into growers that we are going to face another round of extinction, not of bananas. They like to put extinction of bananas, extinction of a particular commercial variety of banana, for sure. But this disease, unfortunately, also affects indigenous bananas and a wide range of local bananas as well. So this really has meant that all effort has been focused onto the banana industry is huge. It's a $30 billion industry worldwide, but they're really a monocrop, and they're really susceptible to attacks like this. But fungal genomes are complex, as we know, and it's not that easy. It's now easy to sequence it now with cheap sequencing, fast sequencing. Even our lab. So, I just wanted to share that this was the genome that we sequenced quite a few years back, and it's in the Fusarium database. But what we really needed was really the next generation analytics that we're able to predict what could be the next emerging pathogen, because it's not enough to just be dealing with the pathogen at hand. And COVID, we've seen this. We have to be prepared, so to speak, for the next pandemic, and that's really what biosecurity is all about. And that's why we're really excited with tools like AI that can keep up with the analytics real time. As people dump sequences into the database, the AI should be continuously mining this and coming up with alerts for us on trends and potential threats, of changes in pathogen behavior and so on. As scientists like us, we keep just dumping data in. So data is good, but what it means is even better. And that brings us to what AI means to agriculture. And it really is the future of agriculture and the future of agriculture biosecurity. So this is from the FAO report on pests and pathogens. And one of the reasons why they say, well, there are strategies now that we can do that we have to put in place. So for pathogens, there's predictive insights, there's disease diagnosis, there's soil monitoring, and many other applications, too much for me to just talk about, but just enough for me to say. So AI, we expect it to be able to detect, determine, predict, and propose the solutions, not just analytics for the laboratory. And the most exciting for me right now, and we'll go through this very quickly, will be the AI-supported point-of-care rapid diagnostics. It sounds like you're in the healthcare talk, right? But this is what we need in agriculture as well. We need to have diagnostics which are readily used in the field. We need it to be used by farmers. We need it to be connected to analytics and experts that can give them solutions and strategies on how to mitigate it in real time. Okay, so that is the future of connected agriculture. So currently, you have to take samples, send it to the lab, you do immunoassays, you do PCR based, or we still have the gold standard of looking at symptoms in the field. But even symptoms in the field now, we can use AI because now we have tools to have disease recognition through phenotypic imaging, and as speaker before this talk about image bank for healthcare, but we really need much more image bank for health of plants, for farmers to be able to cope with new diseases and new threats, especially when we start introducing new crops into the field. So what we're hoping for very soon is field deployable point of care devices, rapid extraction methods, which can be done in the field, and AI assisted pathogen detection. So, there's things that are already in the lab. On the left is like they're using mass spec sensors. I'm sure it's not as big as that. They're like stickers on the plants, mass spec sensors, which can actually do real-time imaging. And then in the market already, of course, we have field-based rapid sequencing techniques like the Minion technology. And then we have also computer vision for diagnosing plant health, which has seen some applications in the field already. So a lot of stuff out there, very exciting. And all of it should be put together into an integrated plan. This is just a really nice surveillance framework that was done in the United States to incorporate AI and data processing into coming up with mitigation strategies. So you get up-to-date data occurrence, disease occurrence data by any means, and then this will be collected from all over the country, and then it will be analyzed, and mitigation strategy will be deployed so that the farmers can then start, or the agriculture system can start to mitigate any serious threats. So this was done for soybean and wheat. Of course, these are big crops in the United States. And you can see the publication last year. Very comprehensive framework that you can set up. And this is really intelligent agriculture and intelligent plant control. And one of the things that computer power really has enabled us to do is prediction. And this is a list, and this is in the climate change review document by FAO. The studies, and there's too few that have attempted to simulate future crop pathogen patterns based on what we are planting, the acreage, the weather changes, and so on. And you can see, of course, most of it are temperate crops, except for banana and rice. And for banana, of course, we are focusing for fusarium wilt, and this was work done due to the invasion of the pathogen in Latin America, which is really where all the big plantations are, and in rice as well. Leaf blight, which is a real threat to rice growing worldwide. And this is a study done in India to look at what will happen in the next 50 years. So very exciting kind of data. And if you're in biosecurity, this is the kind of output that you want from your genome data, incorporated with phenotypic data and incorporated with real data on the field, to give you some sort of a big picture of where you should put your security strategies in place. And
you have 2 minutes.
oh, okay, so I'm pretty late. So I'm not gonna talk the last part. I'm gonna skip this section. So you can, with all this data, you can also do control strategies, and you can have a look at this because it's not new slides, but these are things that we did in the lab with the genomes that we did in our survey to create genome modified plants that were resistant to viruses that were currently circulating in the field. But like I said, we need data constantly to make sure that whatever we develop in molecular breeding or whatever we develop as control strategies is really against currently circulating pathogens. And we did the same for dsRNA. So all these are strategies in the lab that you can do if you have. And I finish off by, of course, now you can do what do you call it? You can do really precision engineering to enable you to create new strategic controls. And this is the use of CRISPR to really modify viral genomes to enable us to control outbreaks. And just within the last 2 years, huge number of papers already out. And this is like half a page only. And the path forward. So I end up with the path forward. I've got 3 slides on this, so bear with me. So the path forward is that, of course, we need data. Just now, Prof. Sarah mentioned, how do we manage open data? We just need it, but we don't need open data in the sense that open data doesn't mean that everybody gets to access it. Open data means that the people who need to access it must have it. So consortium of plant pathologists or people who are working on biosecurity mitigation, they need to have access to this data to enable, cross-border strategies to be made. So it needs to reflect what you need in your local environment, keep it local. So data directly from on-site, on-farm surveillance. We need to accelerate the use of integrated point of care system. We need to network experts. We need to ensure long-term. We need to keep it open. It has to be an open database and curated. And it has to have analytics attached to it. So you need the AI model to constantly work in the background. Then we need to deploy the path forward. And this is where I believe very much in transdisciplinary research, because we need agile and technology responsive regulations. Again, Prof. Siti talked about regulations. So regulations, sometimes we are looking at how to deregulate the process to enable the technology to move faster to farmers. We need to accelerate the acceptance of the new tools. And we did a study in Sebar with Dr. Nozatil. And we looked at the perceptions of our smallholder farmers on using biotech, and it is very poor, because not because they don't want to, but because they are not familiar with what we are trying to offer. So we need to engage earlier. We need to engage more with farmers, because this survey really showed that there's a very low degree of understanding on how biotech can actually help them specifically. And last of all, we need to put it all together. We need to place it in farmers' hands. So we need to give them access to data and technology. We need to connect them. We need to facilitate partnerships to enable this. So we need to, this is a sociological term creeping in, we need to democratize access to information and technology in our field. So it is about understanding what transdisciplinary means. It is about understanding what sociological needs, economic needs is. We need to understand what engineering and IT tools that we can use, and then we can drive the future farm, so to speak. Thank you very much. I hope that give me a little bit of opportunity to share something I feel very strongly about in terms of how we need to move together. And like I said, I was really happy to hear from comments from the speaker and the chair this morning, exactly the thoughts that I had about collaboration, access to technology, open data. Thank you so much.
Yeah, thank you, Prof. Yasmin, for your interesting talks covering a broad aspect in agricultures, like from genomics, data, from plant health, and also covering the biosecurities. So yeah. So to all participants, if you have any questions, please do put your questions in the chat box. So I think I saw one, so maybe we can just quickly take one. So there's questions asking about, like, are there private companies that are actively working in Malaysia today to support your work and of your colleagues?
So sorry, the question was, there's two questions here.
In the field of bananas.
Oh, is that question? Research from the field of banana cited by patents, is it?
Yes.
Oh, yeah, unfortunately, some of the early work by for the detection of some of the viruses were patented, but I believe there's ways around this. A patent is not endless lock, so to speak, and dialogue with industry is really important, because the way that, for instance, the patent on one of the patents that was filed was on BSV, exactly the pathogen, because it was a very serious issue in Cavendish. But the way that all these pathogens are spreading across borders, I think it's in the interest of industry to really have dialogue with developers, with researchers, and with farmers to enable solutions to be created. So, dialogue, interaction, and more space for industry to talk with researchers, I think is really key to resolve this issue of patents. Yeah. So I work in tech transfer the last 5 years, and this is an ongoing problem, to patent or not to patent, and what does a patent actually mean? Patent means you have a control. I mean, CSIRO is the best example of patent and then give open access, right, to a lot of the stuff that they do. There are many ways to look at patents, actually.
Yeah, thank you, Prof. Yasmin, for your answer. So I think it seems that we are run out of time. So perhaps we can proceed to the next speaker. Right. So next we have Dr. Lee Soon Leong from Forest Research Institute Malaysia, FRIM, to talk about forest genetic resources management using DNA technology. So Dr. Lee joined FRIM as research officers in 1995 and promoted to senior research officers in 2001. So currently, Dr. Lee is the head of genetics laboratories, FRIM, and leads a multidisciplinary research team addressing research topics related to conservation, management, and sustainable utilizations of forest genetic resources. So Dr. Lee, you have 20 minutes. So let's welcome Dr. Lee.
Okay, thank you, Dr. Tan. Let me share the slide. Okay, good morning. Good morning, everybody. So I think thank you for inviting me to share some of the activities that we carry out in FRIM towards this management of forest genetic resources. So I think in this talk, I would like to share, I think our emphasis in terms of forest management, in terms of these enforcement of these forest regulation, and also I think related to all these conservation guidelines. So I think here are some of the background in terms of these forest regulation. So illegal harvesting of forest resources poses a significant threat to the sustainability of forest ecosystem. So hence, since 2008, DNA technologies has been developed in FRIM to assist in the enforcement of forest regulation and associated timber threat. So how exactly these DNA technologies have been used or developed actually to assist in these enforcements of forest regulation? You can just imagine it. Let's say this is the suspected timber. It means illegally harvested. So in order, I think, to assist the enforcement, the first thing we need to know actually this belong to what species. Species. For information, I think for Malaysia, we have about more than 400 these common timber species. So without these leaf, all morphological characters, just like actually the stump, how are we going to identify the species? So once we know the species, then the second challenge will be these actually suspect timber actually is originated from where? These I think go to this geographical origin. Is it from northern Malaysia, Peninsular Malaysia, or southern Peninsular Malaysia? And the last, I think the most challenging part is only be possible that we can trace back, these the timber into their original stump. This stump. So in order to answer this question, actually we pose, I think, 4 questions for us to conduct the R&D. So the first one is how to establish this DNA database for species identification. So this one, actually, we are using DNA barcoding technology. Then the second part, how to establish DNA database for population identification. And last, I think, how to establish DNA data for individual ID. So beside that also, is it possible to extract quality DNA from dry wood or wood product? So in order to answer this question, we have used one of the most important timber species in Peninsular Malaysia, that's Cengal, Deobarano helmi, under the family of Dipterocarpaceae. Then we have successfully developed a DNA-based timber tracking system for these important timber species in Malaysia. So start from develop the marker, identify the suitable marker system that can be used for DNA profiling. Also, I think apply all these forensic DNA forensic technology to trace into the original stump. So subsequently, I think this one we spent about 3 years. 3 years, I think, to develop the system for this particular species. Subsequently, I think we continue, I think, to develop for others. So at the moment, actually, we have the databases for 12 important timber species. You can see in this list from some Shorea, Compassia, until some these Rhizophora. Rhizophora is bakau. It's a mangrove species. So in order to build this database, actually we have collected altogether 366 population and using more than 10,000 of these samples. So there are some recently developed Java aromatica and Java oblongipoli. Yeah. So ongoing, we have I think now ongoing develop for another 3 more. So by 2025, I think we will have about 15. Yeah, it's 15 these important timber species that with available of these the DNA database for timber tracking. So beside that also we develop actually for species ID, species identification. We have developed. So just now I mentioned about 400 over important these common timber. So now in our database we have about nearly 700 species. So this one include those lesser known timber, those less common these timber species. So beside that also, I think we have a database also for more than 300 herbal medicinal plant that is been used in Malaysia, and also wild fruit trees. This one I think is important for the improvement of these fruits, and also CITES listed plant species. Those I think critically endangered plant species. In particularly, I think orchid and some Nepenthes, the Nepenthes species. So I'll use, I think one of the good example, Aquilaria malacensis. I think most of you know that Karas is a agawut. Agawut. This particular species actually is a main source of high-grade this agawut in South Asia. I'll use this as a how to explain how actually we develop the species identity database, population identity, and individual identity database. So in terms of species identity database, we have I think in Malaysia we have 7 these most common these Aquilaria species. So you can see in the list. Then we collected the sample. Then based on a 3 chloroplast region, RBCL, MACK, and ITH2, we managed actually to dissolve the 7 species with 100%. 100% dissolve the 100 species, the 7 species. So for the population identity database and individual, the first step actually what we need to do actually we did a very thorough population survey throughout Peninsular Malaysia. So in total actually we have visited 48 these are 4 reserves throughout Peninsular Malaysia in order to find these Aquilaria, these malacensis. So here I think what we observed during our field collection, the major threat, I think the first one will be the illegal harvesting. Illegal harvesting in the forest. These are some of the photos showing that. And second, I think the threats will be on these injury of these tree to look for the gaharu. So you can see the severity of the injured on this individual tree. So these 2 are the major threat of these species in the wild. So from the population survey, we managed to I think identify or collect the samples from 35 these 4 reserves throughout Peninsular Malaysia. From Kedah until Johor, and also include Langkawi Island and Penang Island as well. So based on all these samples, the first thing by using actually these some chloroplast region, so we did this sequencing. Then we managed to come up with this haplotype distribution map. So based on 7 these chloroplast region, then we identified 29 haplotypes derived from 7 chloroplast region throughout Peninsular Malaysia. So in our application, you can see that if let's say we have one these suspected agalut, so what we need to do actually is we just get the haplotype. If the haplotype is P2, then we can zoom actually this most probably come from in the northern region, in the Langkawi area or in the Kedah. So this one I think we will not be able to zoom in into a particular population, more I think toward the region. So the similar, I think, using the same samples also, we have developed altogether 12 microsatellite or short tandem repeats marker to generate the DNA profiling database for these species. So after analyzed, and you can see that in terms of this genetic structure, the Aquila markensis in Peninsular Malaysia basically generally can be divided into two major genetic clusters. Two major genetic clusters. One is the cluster Kedah and Perak, those in Kedah and Perak. The other cluster is Kelantan and Johor. So based on this DNA cluster, so what we did actually, we developed the DNA profiling database based on all these genetic cluster. So you can see that here got two these databases for Aquilaria in Peninsular Malaysia. So for forensic application also, because since we can detect strong genetic structure, also I think in terms of plant, is different from human being. In plant, beside I think outcrossing, the plant also can produce offspring through selfing. So the first step, I think we need to check the conservativeness of these databases. So after that, then we adjust, to make the database conservative. Then after that, then we adjust, for the theta. Theta actually is a population differentiation, these parameter. We adjust, I think, these theta value for both of the databases. So beside that, also these DNA profiling databases can be used, for assign the population. We call it assignment test. So show that the individual can also be used to identify or authenticate source of origin. So based on our simulation study, the mean percentage of individual corrected assigned into origin percent or more than 90%. So that means this DNA profiling database can be used also to identify the population using these self assignment tests. So in terms of application, like for example, these illegal harvested agarwood, we know actually this from this Penang Island. So what we need to do actually is we need to go and collect the stump, the potential stump in the particular for reserve. So if let's say these agarwood originated from this stump, what we will get, in terms of this DNA profile. Here, I think the DNA profile based on the 12 STL loci. So they will have exactly the same DNA profile. So in order to rule out the match due to a random match, so we apply, then we use the database to calculate the profile frequency. Like just now I mentioned, we need to make some correction. This one, I think in the forensic, actually we are applying the product rules. But the product rules with some modification in order to cater for plant that have a strong genetic structure and also, the problem of selfing. So this is, I think, basically this formula is a product rules then adjusted corrected for genetic structure and selfing. So then we calculate the profile frequency is 6.19 times 10 power negative 10. So in terms of random match probability is 1.62. So this also means, so the match due to random match is 1 in 1.62 billion. But normally in the report, we will just highlight in the likelihood ratio, I think for those in the forensic field. So the DNA evidence is 1.62 times 10 power 9 times more likely if the agarwood trunk came from the stump than if the trunk came from the same unknown stump. These are basically, I think, we will highlight in the reports for this forensic report. So we have, I think, various databases. How are we going to manage it? So in order to do that, I think we have developed a management system of this. We call it DNA database management system for forest resources actually in Malaysia. Then you can access, I think, now I think it's still not open. I think soon you will be able to access through this URL. So under this DNA database management system of all resources, basically can be divided into 4 these major modules. The first one is my DNA. These consists of about nearly 26,000 these DNA samples of various plant species from the forest. So it's about 1,000 these 700 plant species. So the second one is my marker. So this one comprises of various type of these markers that have been developed for DNA profiling, for timber tracking, for species identification. So now my barcode. So at the moment, I think in the database consists of these about 700 over these 700 these timber species, about 300 these herbal medicinal plants, and also others these critically endangered plant species. So the last one I think is my track is the DNA tracking this system, this database. So in order for the application also, I think for forensic, we come up with a 4 standard operating procedure. Obviously, you can see that the SOP 1, 2 from the sample collection, DNA isolation, sequencing, and also I think the genotyping of these short tandem repeat or microsatellite. So this is, I think, example of the SOP 1. I think we have developed how actually we collect the sample in the field for forensic investigation. Then emphasis of a chain of custody records, all these have to be sealed. Then we come up with all these kit. Then also once we got all these, then we'll provide the training for enforcement officer. This one enforcement officer is from the 4 department and also from the Wildlife Reserve, the Penghilitan. So far I think we have conducted about now 13 series of this one, the training for these enforcement officers. So in terms of application, all these system now can be used for the first one, I think for enforcement, illegal logging, the wood industry, for herbal industry, food industry, also insurance industry. So I'll just highlight a few. I think for the enforcement, this one application, illegal logging. This one I think happened in 2017. So when these illegal activities have been, I think, happened in one of the 4 reserve, So some of these illegal harvested log have been moved to these sawmill, about 250 kilometers away from the site. Some I think left behind in the site. These are one of the sawmill. Then these are some of the log left behind. Then this is the area. So this one actually is in Selangor, in the Serenda. So actually what we did actually is by using our databases, so we managed actually to track, those illegal harvested log into their original stump. So this one will provide direct forensic evidence to prove that the log has been illegally harvested from the 4 reserve. So under this Serenda 4 reserve, logging license not issued, but they have some logging activities. So once we can match, track back the suspected log into the original stump, you provide a direct forensic evidence to assist actually this enforcement. So these are some of the photo. This is a sawmill. Then this is confiscated this log. Then this is how our team actually made a sample collection. Then our team also a sample collection of the stump. So this is another example actually is involved in one big this chenga tree in this wildlife reserve. So they have confiscated this tree, this chainsaw. Then by using all these databases and also DNA technology, we managed actually to provide evidence that this particular chainsaw have been used to cut down these chenga trees. This one happened actually in Pahang, in one of these wildlife reserve. Then this is how it look like. These are the huge chenga tree that been fell down. Then this is, I think, how actually we have collected the stone dust, from the chainsaw. So also another one I think is very recent on this agar wood. So they claim actually the agar wood actually is from Kelantan, but in fact actually it's not from Kelantan. Actually it's from Malacca, Johor, and Pahang. It's one of the factory in Johor. So also I think in terms of industry, this one I think you use the database also I think to help the dispute or you say a wood for swimming pool. This one I think is one of the construction in the timber company, a dispute between the two this company in Singapore. So the construction company actually they have constructed this swimming pool decking actually using the chenga wood from this timber company. But later on, they found out actually they are not using Giam. So I think they put all these in the court. Then we will provide the DNA evidence. Then actually our DNA evidence show that actually this timber company they are supplying, actually is a real Chengal wood for this construction company. So also I think another company actually they are export all this crewing to the European country union. So now I think for EU, they have a timber regulation. So before they can enter the EU, they need to identify the species. Then we are helping this company actually to identify the growing, the Diptocarpus species. So also I think to testify in the court also, this is how actually we provide the affidavit and we testify in the court. So beside, I think for the enforcement or industry also, all these information also we use for genetic diversity study, in terms of genetic and genomic. So to come out, with the various style of guidelines that can be used, actually to conserve the forest genetic resources. The first one is in situ conservation guidelines. This one basically, I think if you're interested, you can refer to this paper. We are trying actually to answer the question. The first thing, when you talk about in situ conservation, how many populations? So once we have identified the population, how big, each populations. So these are basically very comprehensive type of in situ conservation guidelines. So far, I think we have to generate, I think, more than 10 species in Peninsular Malaysia. So beside that, also for germplasm collection guideline, this one in particularly, I think important for those critically endangered or those threatened plant species. For example, Aquilaria malacensis. So they are going to disappear in a while. So now we need to conduct this germplasm collection. These are the guidelines. So in fact, I think this guideline have been implemented. So the germplasm, I think, have been established, I think, in 2 sites in Peninsular Malaysia. So another one is a plant material transfer guideline. So you're interested also, I think this online, you can refer to our book actually. Basically how actually, if they say you want to do a replanting, how are you going, where we need to collect the plant material. This is a plant material transfer guideline. So in conclusion, DNA techniques are useful tool to assist in the enforcement of forest regulation, associated trade and industry, and in conservation of these forest resources. So we are putting our effort to complete the databases with majority of important plant species in Malaysia and to establish the National Plant DNA Forensic Center to assist in illegal logging enforcement and promoting trade in a legal forest product and to develop various conservation and management guidelines to strike a balance between conservation and utilization of forest genetic resources. So with that, thank you for your attention.
Yeah, thank you totally for sharing your interesting talk about how we can actually preserve, or manage our forest genetics resources and everything. So I think we are run out of time. So perhaps participant, if you have any questions, you can just post your questions in the chat box. I'm sure Dr. Lee is very happy to answer your questions. So next, we move on to our next speaker. So our next invited speaker is Dr. Win Siu Ng from Malaysian Palm Oil Board, MPOB. So Dr. Win obtained her PhD from University Putra Malaysia before joining the Malaysian Palm Oil Board as research officer. So she has nearly 20 years experience with MPOB and is currently leading the epigenetics team in the Advanced Biotechnologies and Breeding Centers at MPOB. So, Dr. Ui, the screen is yours.
Thank you. Thank you, Dr. Tang, for the introduction. Let me share screen. Okay. So you can see my screen, right?
Yes, but could you put it in a presentation mode?
Okay.
Yeah, good, go.
Okay, I also like to thank the organizers for giving me this opportunity to share this research with you. For this study, we looked for transcriptome signatures in the mother palms itself that can be linked to their embryonic outcomes. All right, this is the outline of my presentation. Okay, first I'll touch a bit on oil palm productivity and then on the oil palm tissue culture cloning process itself. Before we started this study, we also had to assess the embryogenesis reproducibility from the same mother palms because we, due to the sampling method that we are going to conduct for the transcriptomic study. So after the transcriptomic analysis, we identified some differentially expressed genes, DEGs, okay, that were associated with highly embryonic otets. And we are currently working on some prediction models for embryogenesis. So this is a chart that shows you the productivity of oil crops that produce our common cooking oils. As you can see here, oil palm is the most productive oil crop. It contributes about 43% of the world's edible oil, but is planted on just 5% of the land that is occupied by oil crops. Okay. And so with the shrinking agricultural land, there are several upstream strategies to increase the productivity of oil palm. First one is to continue with replanting program. We have to replant the palms that are old because they are very tall and that makes harvesting very difficult. So we need to continue with actively replanting of this kind of palms. Second strategy is to ensure production of quality and high yielding planting materials, mainly through the breeding programs and tissue culture programs. And then third is to speed up the adoption of automation and mechanization, and especially in innovating better machines for harvesting and management of the plantations. And fourth is to strengthen the management of pests and diseases on the oil palm. So for this particular study, because we are looking into trying to improve the efficiency of the tissue culture process, we are working towards contributing to this strategy to produce good yielding planting materials. Okay, so the oil palm tissue culture process starts with the sampling of leaf cabbage. Okay, this is the leaf cabbage, what we call the leaf cabbage. So the leaf cabbage consists of young leaf tissues that are used as explant tissues to initiate the tissue culture process. Okay, this process undergoes what is called indirect somatic embryogenesis, which needs the intermediate callousing phase. So from this callus phase, embryonic callus, callus that are embryonic will spontaneously emerge and then continue to develop into somatic embryos and then multiply further into polyembryons, whereby we will then initiate shoots and roots, and finally we will get the tissue culture plantlets, which we call ramets. Okay, this entire whole process takes about 5 years for the oil palm, so it's quite tedious. So the main bottleneck for tissue culture is the embryogenesis rate, and coupled with the short supply of the Ternera otets. Ternera is the commercial hybrid planting material in that we use for oil palm in the oil palm industry. So the short supply of the Ternera otets, the low embryogenesis rate, and the uncertainty of the embryo culture's development, all these issues have hampered the efficiency of the tissue culture process. So generally, in plants, somatic embryogenesis induction is preceded by the transcription of many genes that are related to stress. And one of the factors that influence somatic embryogenesis is the physiological state of the mother plant. So we ask the question, can the cellular and physiological state of oil palm otets that we will examine here using transcriptomics, can this be associated with its embryogenic potential? Okay, but before we move into that, we had to assess the reproducibility of embryogenesis from the same mother palms. Why? Because we will be conducting sampling of the leaf of the samples for this transcriptomic study from selected otets that we already know the embryogenesis outcome. That means they had been cloned before. So we had to assess whether embryogenesis is reproducible, because when we sample the tissues for transcriptomics, we will not be conducting clonal propagation again because it will be too expensive. Okay, so tissue culture industry do conduct this what is called resampling. That means they go back and resample the same mother plant for embryogenesis for tissue culture. So we collected this kind of data from the industry and we plotted this is embryogenesis outcome from the first sampling versus the embryogenesis outcome from the second sampling. So the data shows you that there is some correlation pattern that you see. We measured this using what is called concordance correlation coefficient analysis, okay, which is a method to measure how far the data points deviate from the line of perfect concordance, which is the y equals x line. So the concordance correlation coefficient we measured here was 0.66, which only indicates a moderate concordance, still is concordant. But alternatively, we can look at the data based by categorizing the data into two categories here, non-embryogenic and embryogenic outcomes. So for example, this 22 means 22 otets that were non-embryogenic at the first sampling were non-embryogenic again at the second sampling, so they are reproducible. So to count the reproducibility here, we count the numbers in the black boxes, and we got about 82% reproducible. If we split the embryogenesis categories further into two categories, low and high embryogenesis groups, we get about 72% reproducible, which is still generally quite high, quite a good reproducibility. So with this in the back of my mind, we continue to generate the transcriptomes. So initially, we worked with 3 tissue culture labs on this project. Initially, we had wanted to generate transcriptomes from the leaf explant itself, because these 2 tissue culture labs, A and C, had freezers. So they could store a portion of their explants in the freezer when they conduct tissue culture of the otets. Okay, but tissue culture lab B did not have such storage facilities, so they sampled the spear leaf for us instead from the selected otets. So because samples were limited, we just generate transcriptome from all these tissues. Okay, so looking at the principal component analysis of the transcriptomes, so each data here or each point here represents one otet. So for tissue culture lab B, where we use spear leaf tissues, the red data points here are actually belonging to 0 and low embryogenesis group. Okay, and the blue one is associated with high embryogenesis outcomes. So you can see from the tissue culture lab B samples that they are clustering according to the embryogenic outcomes. So this was quite interesting. But for tissue culture lab C and A, where we use cabbage samples, you see there's a big overlap in the 2 embryogenesis groups, the non-embryogenic and the embryogenic groups. And if we integrate all these data together, we see that the variation between the 2 types of leaf tissue, okay, cabbage and spear leaf, is very, very big. There's also some variation between the tissue culture labs, and all this variance is much bigger than the variance between the embryogenesis groups per tissue culture lab. Okay, but we could not ignore this clustering profile, which was already looking very interesting. So we just decided from this to go back to sample the spear leaf instead from tissue culture lab C and A. But unfortunately, only tissue culture lab A could provide spear leaf samples. So we proceeded with samples from tissue culture lab B and A. So transcriptomes from the spear leaf shows that the clustering is again based on the tissue culture labs itself. Okay, the variance between the tissue culture labs are much bigger than between the embryogenesis groups. So we decided from this profile to conduct the differential expression analysis separately for each lab. Okay, so for tissue culture lab B samples, you see that we categorize them into 3 categories: 0, low, and high embryogenesis, based on the number of embryonic lines. Okay, and you can see here that the 0 embryogenesis group and the low embryogenesis group overlaps. but the high embryogenesis group forms a distinct clustering by itself. Okay, and if we do differential expression analysis between this group and this group, we get a few thousand DEGs or differentially expressed genes. But for tissue culture lab A samples, even though we're using spear leaves, we see a big overlap between the 0 and low groups. Okay, for tissue culture lab A, we do not have OTATs that are belonging to the high embryogenesis groups because there were no OTATs that fit into that criteria. That means more than 90 embroid lines. But the advantage for tissue culture lab A samples is that there are a lot more observations per embryogenesis group, a lot more samples per embryogenesis group. So we did differential expression analysis, even taking into account the genotype factor here, and we identified about 170 differentially expressed genes. But then the question became, how do we integrate the results from tissue culture lab B and A? So what happened was that we decided we would take these 171 DGs, pull out the expression values from the transcriptomes of tissue culture lab B and see how the clustering looks like. So we see that based on the expression of the 171 DEGs in tissue culture lab B samples, we see that the OTATs are still clustering according to the embryogenesis outcomes. So this is the high embryogenesis group, and this is the 0 and low embryogenesis group. You can also see the clustering is obvious from the expression heat map here for the 170 DEGs. The high cluster here, one cluster, and another cluster here. Okay, so from these results, we continue to focus on these 171 DEGs. So based on the functional annotation of these DEGs, we see that quite a number of them were involved in these functional categories: flowering time and reproduction. So these are some of the genes that are involved. Light or photosynthesis related, immunity and stress response, and nitrogen uptake and metabolism. So the genes highlighted in red here, for example, FRL. FRL is actually Frigida-like genes, which is involved in flowering time. So there were 3 members of the Frigida-like genes that were identified as differentially expressed. Okay, and similarly here, there are 2 GHD genes that were identified as differentially expressed. Incidentally, there are only 2 GHD genes in the OIPAM genome that we found from the NCBI database. Okay, so from this, we moved on to qPCR verification. So for qPCR verification, we included some samples that we use for RNA-seq, but additionally, we included a lot more new OTAT samples. Okay, so we took about, I think 21 RNA-seq samples, and we included about 31 new OTAT samples. So as you can see, based on the expression profiles from the qPCR for per embryogenesis group, this is 0, low, and high. This column of genes are those that are involved in the flowering time and reproduction. You see and then this column here is related to light and photosynthesis. You can see the profiles are quite similar, and these are the genes involved in the other pathways. Okay, if I were to zoom in into the FTIP gene, which is FT interacting protein gene, the mean expression in high embryogenesis group is significantly different from 0 embryogenesis group, but it's not significantly different between low and 0 or low and high. So most likely because of the wide range in expression per embryogenesis group, and we see this wide range of expression in most of the other genes as well. So from here, we ask the question whether we can use a combination of the genes to associate with embryogenesis outcomes. So at this point in the study, we met a group of data scientists from University Malaya. So we had some discussions with them, and they proposed or suggested the use of what is called a linear discriminant analysis method. So the linear discriminant method on just the expression of the 37 DEGs from using qPCR data can actually discriminate the 0 and low embryogenesis groups better. If you remember in the earlier slide where the zero and the low embryogenesis group usually overlap, especially for samples from tissue culture lab A. Okay, but linear discriminant analysis or LDA can discriminate these two groups, regardless whether they're coming from tissue culture lab A or tissue culture lab B samples. Okay. So along the study, then after that, we optimize some of the LDA method using a smaller set of DEGs from the 37 DEGs. We see that even using up to 15 DEGs, we can still discriminate the zero embryogenic group from the other two embryogenic groups. Okay, but this LDA at this point of time was trained on a set of 58 samples. Okay, and we did not have any new samples that time. So along the period of the study, we got more and more new OTAT samples, which we generated some more qPCR data, but we didn't generate qPCR data on for all the 37 DGs, just for a subset. And we tested this out also with 10 new OTAT samples. So we also tried another algorithm called support vector machine. And we see here that the support vector machine seems to perform better in terms of prediction for the new OTAT samples compared to LDA. Okay. So we started off with 13 DEGs, training for 13 DEGs, and we also use a subset of these 13 DEGs, and also 12 and 8. So you see that the model with the 12 DEGs for SVM predicts better, 100% correct for the 10 new samples. It has 100% sensitivity in predicting true embryogenic outcomes, and an 84% specificity in predicting non-embryogenic outcomes. So we are still optimizing these models or the DEGs. And how would we use such an assay, and let's say in the for oil palm tissue culture sampling? So in practice, the tissue culturists, after identifying the OTATs or selecting the OTATs for tissue culture, they will go and sample the leaf cabbage. But if we were to use this predictive assay, we propose that you would first go and sample the spear leaf, just the spear leaf. So the advantage here is that sampling of the spear leaf did not harm the plant compared to sampling of the leaf cabbage. Okay, and then you conduct the predictive assay and then predict which embryogenic outcomes it might potentially give, and then only go and sample the OTAT. So this is my last slide. I'd like to acknowledge the technical support from our breeding and tissue culture and bioinformatics units from MPOB, my two collaborators, AAR and United Plantations, and of course the invaluable advice from the team from University of Malaya Center for Data Analytics. Thank you.
Thank you very much, Dr. Ui, for sharing her research with us on these oil palm leaf transcriptomes. Let me quickly check. Is there any questions in the chat? I was disconnected, so I can't see. Is there any question posted just now? But anyway, I do have one question.
Yes.
Perhaps, no questions. Okay. Yes. Thank you. Yes. Dr. Ui, perhaps I missed something when you talk about using the spare leaf for your transcriptome. Perhaps could you tell me what is the rationale of using this spare leaf for as a
okay.
sample?
Yes, that was also my initial hesitancy. So the thing is, because we wanted a tissue that was easily accessible, and also because we just wanted to see whether we can associate the health of the physiological state of the palm. So using the leaf tissues, which is abundantly available, also easily accessible, we thought we would try and see whether there is any transcriptomes that we can associate. And from this study, we see that there probably is. So just the leaf tissues itself may be good enough.
Yeah. Okay. Right. Thank you. So to all participants, if you have any further questions, you would like to ask Dr. Uy and our previous speaker, Dr. Lee, and also Prof. Yasmin. So you also can post it in the chat box so that we can compile and then send it to our speaker. Right, so I think it marks the end of these sessions, and now we will have a 5-minute short break. So please do join us back at 11:30 a.m. See you.
Thank you.
Okay, all right. Good morning, everyone. It's time for oral presentations. This is concurrent session 2. So I am Dr. Bhaskaran Gunasekaran from UCSI University, and I'm the chair for oral presentation in Hall 1, which is for agriculture, aquaculture, biotechnology, and molecular biology. All right, so I would like to remind all the presenters that you are given 10 minutes to share your work, right? And at the 9th minute, you will hear the first bell, like this, right? This is just to inform you that you have another 1 minute to wrap up your presentation. And at the 10th minute, you will hear the second bell, like this, 2 times. Okay, this is just to remind you that you have reached 10 minutes of your presentation and you can settle down. Right, so all presenters, you're reminded to stay in this hall, okay, till the end of the session, because at the end we will have 15 minutes Q&A. Okay, so participants, if you have any questions to the speakers, you can put the questions in the chat box, or you can raise your hand up, okay, virtually, okay, for us to moderate you to ask the questions during the Q&A. Okay, be reminded that this session is from 11:30 until 1 p.m. Okay, so let us start with the first presenter for today. Let me welcome Dr. Izwan Baharudin from University Kebangsaan Malaysia. So he's going to talk about understanding the oil palm pathogen Scleroderma boninensis from multi-omic studies. Dr. Izwan, are you here?
Yes.
Yes, you can start.
All right. Let me share this.
You may share your slides. All right, I can see.
Okay.
You can start.
All right. Thank you, Mr. Chairperson, Dr. Bhaskaran. Okay, I'm Izwan from University Kebangsaan Malaysia. And today I will be presenting the research study in our lab, which is on the topics of understanding the oil palm pathogen of Scleroderma boninensis from multi-omic studies. So I think this one has been mentioned by our previous speaker, Dr. Uy, which is the palm oil is one of the most important oil producing crops in the world, as it contributes about 31% of the world vegetable oil supply. In addition to that, so actually it only requires about 0.26 hectares of land to produce about 1 ton of oil, whereby the other plant crops such as the rapeseed, the sunflower, and the soybean, which needed more than 1 hectare of land to produce the same volume of the oil. And palm oil is planted in the tropical region, including Africa, Asia, and South America. However, we can see here the majority of it was planted in Southeast Asia, mainly in Indonesia and Malaysia. And actually, both countries contributed more than 80% of the global oil palm production. However, the oil palm plantation in Southeast Asia are heavily affected by a disease known as a basal stem rot, or known as BSR, which mainly causes by three pathogens, which is the Ganoderma boninens, Ganoderma zonatum, and Ganoderma minetositum. However, previous studies have shown that the Ganoderma boninens is the main culprit for the BSR. And actually, the BSR can reduce the weight and the number of the fresh fruit bunch in the still living palm, which eventually will cause a major economic losses for Malaysia and Indonesia, which estimated of losses of up to 365 million US dollar per year. And to date, so there is no methods that can prevent the continuing spread of the Ganoderma disease in the field. And basically, this is the general overview of how the Ganoderma, the life cycle of Ganoderma boninens. So basically, they are needed by two compatible monokaryon needs to be fused and form a dikaryotic mycelia before it can colonize and form the basidiocarp to do the infections of the oil palm tree. And previous study has revealed that the monokaryotic mycelia of the Ganoderma boninens were unable to infect the oil palm tree, whereas the dikaryotic mycelia can. Thus, we find it very interesting to know how the fungus control its strategy for the infection and the survival in the oil palm plantation. So at the moment, there are 4 different genomic data sets that currently available in the public databases, and all strains actually were sequenced that are isolated from different regions in Southeast Asia. As we can see here, 2 strains, which is the NJ3 and G3, which is isolated from North Sumatra. And one of it, the FGVM strain, was isolated from the Serting Hilir in Peninsular Malaysia, and one is from Bium's strain, which is isolated from Borneo. And in addition to that, so there are few transcriptomic databases were available online. As you can see here, our group is also managed to generate the transcriptome data from 3 different growth stages of Ganoderma, which is the monokaryon, the dikaryon, and the mating junction. And in addition to that, other research group was also published several transcriptomic data, but then they are different. Mainly they're performing the transcriptomic data from the interaction studies between the pathogen and the host. So it was postulated that the Ganoderma boninens possess a tetrapolar mating system, which is similar to other basidiomycete fungus. And actually, the mating type loci, or the mating A, which is, or we call it as a mate A locus, is mainly consists of the HD1, or the homoiodomain protein 1, and HD2, which is the homoiodomain protein 2. And basically, both generally the HD genes is accompanied or flanked by 2 different genes, which is the mitochondrial intermediate peptidase, or MIP gene, and the beta flanking gene. And based on our analysis, we managed to identify that the HD1 and HD2 are flanked with MIP and the beta flanking gene in context or 56 in the genomic database. And when we compare whether the MET A locus in our Ganoderma boninens G3 is whether it's similar to other Ganoderma species that being published, then we compare it, and it seems that all of the Ganoderma species share a similar pattern of mating Met-A locus. And when we check at the sequence data of the HD1 and HD2, actually both HD1 and HD2 are conserved between all the Ganoderma species, but we still can see some of the variation in terms of the sequences. And perhaps we postulated that these differences are mainly strain related, not the species related difference. And then we try to find the Met-B locus of the Ganoderma boninens genome, and basically this Met-B locus mainly consists of the pheromone receptor and the pheromone precursor. And one of the pheromone important pheromone receptor protein is the ST3 gene, the ST3 protein, which is the transmembrane protein. And actually, there are two types of ST3 type receptors, which is the non-mating ST3 and the mating ST3 pheromone receptors protein. And most importantly, one of the unique features of ST3 is for the mating ST3 is they must be located within the mating loci and must be close proximity to the pheromone precursors. And our analysis have identified that there are 9 gene encoding ST3 from the genomic database and 11 gene encoding the ST3 genes in the transcriptomic database. And in terms of the pheromone precursors, we have do the analysis, and actually the pheromone precursors which involve in the mating must be located within the mating B locus. And in addition to that, they have a specific or unique features of the pheromone precursors, which they have the specific motif of MDXF at the N-terminal domain and the CAX at the C-terminal domain. And based on our analysis, we successfully identify about 40 gene encoding the pheromone precursors in the transcriptomic and about 22 from the genomic databases. And then we align the genes that we have, and then we have successfully identified that there are 2 potential mating B locus in the Ganoderma boninens genome, which is in the backbone of 53 and 190. So as a conclusion, various omics analysis that have been carried on was to support the fundamental studies of the Ganoderma boninens. So especially in finding the genes that are responsible for the matings in this fungus. And we have confirmed that the Ganoderma boninens has a tetrapolar mating system, and that we think this is the reason why it is quite how to say that impossible for us to combat the BSR disease in the oil palm plantation, because this tetrapolar mating system will increase the diversity of the genetic pool of this fungus. And I would like to thank Universiti Kebangsaan Malaysia for providing the grants, and our research group, the Molecular Mycology Research Group in UKM, and our collaborating partners from FGV, MGVI, and MPOB. I think with that, thank you.
Okay, all right. Thank you, Dr. Ikwan, for your presentation. I don't have the job to ring the bell. It is perfectly on time. All right. Thank you so much. Kindly stay tuned in this call because we will have Q&A session at the end. Right, participants, if you have any question for Dr. Ikwan, you may post it in the chat box. Okay, moving on next, I shall invite Ms. Siti Dalila Muas. She's from ICMBB Plantation Technology, Malaysia. All right, her project presentation is on DNA fingerprinting of commercial madak coconut production and genetic purity testing. Ms. Siti, you can start to share your slides. And you can start whenever you're ready.
Okay. Yep. Okay. Lama tu kedua ni. Okay. Give me a second. Okay.
Your slide is in the display mode.
Okay.
Yeah, can you
Is it fine now? Okay, hi everyone. So today I will be sharing with you my presentation entitled DNA fingerprinting of commercial mata coconut production and genetic purity testing. Before we go into the whole molecular part of the topic, I would like to introduce you to mata coconut and why it is so important. Basically, mata is a coconut hybrid that we get when we pollinate a Malayan dwarf mother palm with pollen from a tall coconut called taknanan. The hybrid is popular among coconut planters due to some of its superior characteristics compared to other coconut varieties or hybrid as shown here. They have early maturity, they have produced more and larger fruit, and also they have thicker copra, which translated to high copra production. And lastly, due to the large nut size and thicker copra, they are considered as a versatile type of coconut, meaning they can be planted to be used for their water, milk, and also desiccated coconut. However, mata production comes with some challenges as in any other hybrid crops production. Firstly, because they are hybrid, we have to make sure the parents are actually pure varieties so that the crossing would not be illegit. Secondly, the mother palms need to be emasculated every time before pollination. You can see in this photo that the male and female flowers of coconut are located closely together in the same inflorescence. And both types are actually naturally self-pollinating because the male and female flowers tend to anticize simultaneously. So to prevent the mother palms from being pollinated by its own pollen, the male flowers need to be removed completely. And the emasculation process is actually very laborious and tedious. So sometimes it can be incomplete, which will lead to contamination due to the mother palm self-pollinating. And because of that, it is a standard procedure to do selection to remove the contaminants during nursery stage, which will require highly experienced operator. And so ultimately, the challenge is actually to produce high purity mata seedlings. So how hard it is to make sure only pure mata are being sold to customers. Let us look at some pictures. As I explained earlier, mata is a hybrid derived from crossing of Malayan yellow or Malayan red dwarf with taknanan tall. In ideal situation, this cross would produce 100% pure mata dividing into green, gold and orange color following the color of taknanan parents as shown here. But in the event of wrong parental palms varieties and very common incomplete emasculation, they're bound to be contaminations such as MYD and MRD seedlings through self-pollinating of the dwarf mother palms. That's why after harvesting and growing the seeds in nursery for a few months, we will select the seedlings to sell based on the petiole color of the seedlings, as you can see here. First, the green and gold petiole color seedlings will be selected as pure mata. And since the orange mata and MRD has a very similar petiole color, usually the breeders will exclude all orange colored seedlings from being sold. In addition, they also remove all yellow seedlings which are supposed to be MYD contaminants. So this eye selection approach alone has shown that it is prone to human error and can lead to overculling and underculling. Therefore, we tried to find a way to support or improve the quality of our mata purity, which has led us to embark on DNA markers development and application for coconut. So why DNA markers? Actually, the recent assembly of coconut genome in 2019 is the main factors since it enables the discovery of single nucleotide polymorphism or SNP. These figures just to show you visualization of coconut genome and its features taken from a paper by researcher from China in CRAD. And then coconut is actually possess a diploid genome with 16 pairs of chromosome. And this is the CRAD database website. where we can gather all sequence information for the markers development. Other than that, ICMBB Plantation also has established an in-house high throughput genotyping platform called SNPline since 2015, which is currently catering to our oil palm marker screening for various purposes. So this ready to be used platform also provides opportunity for coconut markers development and its application. For the method, it is a standard marker development method. We first took 1366 SNP markers based on 4 centimorgan interval from CIRAD linkage map. This is so that we can have good coverage of the whole genome. And then we gathered the sequence information from CIRAD and design them from CIRAD database and design them as probe for 6 SNP, a targeted GBS platform. After that, 768 probes designed that have high good amplifiability probes were selected to be used in the genotyping later. At the same time, we sampled the study population, which consists of 192 coconut samples as described here. Okay, we include all the 3 colors of mata from nursery. We also include all mata parental palms from our parental palms field in Banting. Then we extracted DNA and checked the quality to make sure only high quality DNA being proceeded for genotyping. Then genotyping by sequencing was done on 6 SNP platform at LGC Berlin. After that, when we received the genotyping data, we checked the quality of the data and filtered the markers based on the criteria here. Lastly, we ran the analysis to interpret the data. Okay, for the result, this is the marker statistic after we did some QC on the genotyping data. We have removed those markers with core rate less than 95% because they can be unreliable. Those that are monomorphic as they are not informative, and those that have minor allele frequency less than 0.01 to avoid markers carrying rare allele. The final number of markers is 697 and 192 individuals all have good genotyping rate of more than 99%. So the data was subjected to PCA analysis and plotted into a scatter plot to see the genetic stratification. Okay, each point here represents a sample and the color are as indicated in the legend here. There are 3 main clusters formed in the plot with mata clusters located in between Taknanan and dwarf cluster, confirming that mata is hybrid of the 2 parental palms. We can also see for mata orange, most of them are assigned into MRD and MRD cluster, which agreeing to our prediction of them being the MRD contaminants. And we can also see that there are several individuals that deviated from the main cluster region, suggesting they are the contaminants. Okay, but to further strengthen the result, phylogenetic tree was constructed for the samples as shown here. Unlike PCA, 4 main clusters are clearly formed in the phylogenetic tree. Because the MYD and MRD forming their own clusters instead of combined in one cluster. There are also few unknown clusters formed among the main clusters, and we found that the individuals in the unknown clusters are emphasized in the red line, actually the same individuals as the circle ones in the PCA plot. So we are confident to conclude them as contaminants. Okay, table here is the summary of the cluster based on the tree. The green color percentage are the samples that are assigned correctly into their own cluster, while the rest are of types or contaminants. And then 2 interesting things to see here. First, orange mata are mostly assigned to MRD cluster. Which is confirming our PCA finding that they are actually MRD contaminants from MRD self-pollination. However, there is still 10% of mata orange are assigned in mata cluster, which suggesting overculling can happen and causing losses. Another thing worth to note is the green mata percentage here, which showed that 40% of them are actually contaminants, which suggesting that there was overestimation of purity through color selection alone. I think this emphasizes more on the need of marker screening on top of the color selection by eyes. Okay, so the result has proven that the markers are able to differentiate the varieties.
Right, Ms. Siti, can you make the conclusion ready?
Okay, Next, so we're able to identify the contaminant. So we decide to apply the markers by reducing the numbers of markers to 125 only. So some of the application are we have screen all our active mata parental pumps, and also we have done mata routine screening, which we managed to observe purity level of 92 to 98% every time. Okay, so this is the population structure that we use for the analysis, just to visualize the larger sample size. So for the conclusion, genetic stratification of MYD, MRD, Tanhanan, and MATA using the 125 informative markers as reference, able to identify the contaminants. The DNA markers can increase accuracy of selection of pure mata seedlings, and ICMBB plantation has started DNA markers deployment for mata production. So moving forward, we can expand the markers application to other coconut varieties, and also we can do the QTL discovery, for example, in markers assisted breeding. Thank you.
Okay, thank you, Ms. Siti, for your presentation. And stay tuned in this hall for Q&A session later. All right, we shall move on with the third presenter in this hall. Let me call Mr. Aminu Salisu from University of Science Malaysia. Mr. Aminu, are you here?
Hello, good morning. Can you hear me? Hello.
Yes, I can hear you. You can start to share your slides.
Okay.
Mr. Amino is going to share on bacterial diversity of reservoir soil of Heptris vittata growing in arsenic-rich and natural mineral soils. Yes, I can see your slides. Can you put in presentation mode?
Is it okay? The slide okay?
You can start.
Okay.
Yes.
Hello and good morning, all. I'm going to present. I will not share my research on the topic bacterial diversity of rhizosphere soils of Heptris vittata growing in arsenic-rich and natural mineral soils. The presentation is going to be based on the flow, and I will start with the introduction. Soil is a very dynamic ecosystem. It is essential for the survival of various microorganisms. Arsenic pollution can greatly disrupt the microbial activity in the soil. The microbial communities in the soil are extremely diverse, with 1 gram of soil containing thousands of microorganisms, or thousands of bacteria and fungi. Industrial activities such as mining, tanning, smelting, fence industries can contribute in polluting the soil with arsenic and other heavy metals, leading to soil contamination as well as water contamination. Heptris vittata, also called Chinese breadfruit, can grow in the polluted environment, when the polluted soil specifically, due to its ability to accumulate high level of arsenic into the tissue, into its tissue above ground. So based on this, Heptris vittata was selected, and the soils were collected from the root of these plants in order to detect the diversity of this microorganism in the rhizosphere soil. There are different sources of this arsenic metal, which can be natural or anthropogenic sources. The natural may be, it may include volcanic eruption, hot spring discharge, weathering of rocks, etc. While the anthropogenic or the artificial sources may be from tanning, fence, chemical industries, mining industries. Maybe other sources may be agricultural, fertilizer, cattle waste, irrigation water, herbicide, pesticide, and fungicides, while other sources may include the mining of metals, municipal solid waste discharge, smelting, and sewage water. All this can contribute into the polluting the soil with different arsenic. The objective of this research was to actually determine the bacterial diversity of the soils collected from the rhizosphere of this terrace without plant and compare the population of this arsenic tolerant bacteria in arsenic rich as well as the natural mineral soils. So this was carried out through the 16S amplicon sequencing. Soils were collected from the sampling site, both in arsenic rich and natural mineral soils, and then the soils were transported in an aseptic manner into the laboratory for DNA isolation. DNA was isolated using genomic DNA bacterial soil extraction kit, RBC, Rio Biotech high yield RBC kit for the isolation was used. The bacterial diversity in arsenic rich and natural mineral soils was determined through the 16S amplicon sequencing using high throughput sequencing technology. The B3, B4 region was amplified using local specific sequence primers with overhang adapters. And the stage, during the first stage of the library construction, the flow here was used, yes, this flow was used for the 16S amplicon sequencing, 16S amplicon sequencing streamline process flow using the MySec platform, Illumina platform. This is the summary of the platform of the process. So on the other side, the samples were also analyzed physicochemically to determine the soil nutrient analysis as well as the heavy metal content of the soil. So the soil nutrient, heavy metals, and arsenic speciation to determine the different species of arsenic in the soil so as to ascertain the level of the contamination by the heavy metals was also carried out. Results and discussion. The results shows that the physicochemical analysis of the soil sample showed that the pH of the sampling site, the RH, that is arsenic rich, was observed to be slightly alkaline, while that of the USM was observed to be slightly acidic. So the variation in pH may also contribute in the diversity of this bacteria in the rhizosphere soils. The USM soil was also observed to have higher organic matter content, high carbon content, carbon nitrogen content, as well as the what is when compared to RHT. The RHT have less organic matter, less carbon nitrogen content, and it has higher phosphorus content and electrical conductivity. So this is the soil analysis showing different parameters analyzed as well as the textile classes of the soil, which can also contribute in terms of the soil microbial diversity in the soil. Heavy metal analysis, table 2 shows the heavy metal analysis. From the result, it was observed that the total arsenic content in arsenic-rich soil was observed to be 101 milligram per kilogram to 3,160 milligram per kilogram in the arsenic-contaminated soils, that is arsenic-rich soil of RHT, while in the USM natural mineral soils, the content was observed to be low, 5 to 6 milligram per kilogram. Other heavy metals such as cadmium, chromium, copper, lead, mercury, nickel, zinc were also detected, and the RHT was observed to have higher copper content, lead, and nickel in addition to the high arsenic content, while USM was observed to have higher cadmium, and chromium, and zinc content compared to the arsenic-rich soil. On the other hand, the arsenic speciation of the soil also reveals that the total arsenic content was similar in the heavy metal analysis, while the species, the arsenic 3, which is more toxic, was observed to be higher in the RHG as usual compared to the USM soil. Then arsenic 5, which is less toxic, was also observed to be higher, ranging 2,310 milligram to 92 milligram per kilogram in RHD, while in USM it's around 2.6 to 4 milligram per kilogram. So the phylum level bacterial diversity in arsenic-rich natural mineral and natural mineral soils of rhizosphere cells shows that the top 10 most abundant phyla in arsenic-rich soil RHT was observed to be actinobacteria, proteobacteria, chloroplexi, acidobacteria, that is in descending order. Actinobacteria was observed to be higher than proteobacteria, chloroplexi, followed by acidobacteria, lentomycete, baricomicrobia, bacteroides, and the least one was the Firmicutes. So this shows the phylum level diversity, and the figure 3 shows the genus level bacterial diversity in arsenic-rich and natural mineral soils, with the Pseudoatrobacter having the higher content, followed by Gala, Springomonas, Nocardioids, Pedicomicrobium, and the list was Lysobacter. So this shows that the most abundant phyla.
Yeah, Mr. Amino, you have 1 minute to wrap up your presentation.
Okay, in conclusion, the RHG soils were observed to be richer in species but less diverse than the USL soil. The study reveals that many intricate bacterial taxa with actinobacteria, proteo, and these Firmicutes are the most abundant in arsenic-rich soils. Acidobacteria, Microbia, Lentomyces were observed to be more prevalent in USM. So genes also predicted to be of plant growth promotion and arsenic tolerance were observed in both study sites, indicating that these genes were responsible for the survival of these bacterial phyla in arsenic-rich, and can also improve the arsenic bioremediation ability of the arsenic bioremediation ability and increase bacterial species in the 2 locations. I would like to acknowledge University Science Malaysia for providing facilities for this research. As well, I would also like to acknowledge TED Fund, Tertiary Education Trust Fund, Nigeria, for PhD sponsorship. Thank you very much for listening.
All right. Thank you, Mr. Amino, for your presentation. Kindly stay tuned in this hall. Later, we have Q&A session for you. And for participants, I would like to remind again, if you have any question for the presenters, kindly put it in your chat box. All right. Moving on next, I would like to call upon Ms. Ong Chin Yin from University Tunku Abdul Rahman, Malaysia. Ms.
Hello.
Chin, are you here?
Yeah, I'm sharing the screen now.
Good. So her presentation will be on are flowers attractive to the insect parasitoid Hydrobius imperius of the oil palm batworm, Metisa plana. So she's going to answer this question through her presentation.
Am I sharing my slide now?
Yes, I can see very clear. You can start.
All right. Okay. So good afternoon, everyone. My name is Ong Chin Yin, and the title that I would like to present is are the flower attractive to the insect parasitoid Hydrobius imperius of the oil palm batworm, Metisa plana. So usually flowers are recommended in the oil palm plantation to attract the beneficial flower insect. Which plant does the parasitoid prefer? So usually the flower will emit the organic compounds like aliphatic, benzonoid, phenylpropanoid, mono, and sesquiterpenes to attract the insect. So for the flower, they will act as a food source to attract the parasitoid by releasing the volatile organic compound. So what is parasitoid? Parasitoid are the small insects that will not either attach or just a moment.
All right, so parasitoid are the small insect whose immature stage develop either within or attached to the outside of the other insects referred to as a whole. So as I mentioned just now, the beneficial flowers not only can provide the nectar and also shelter to them and also enhance their longevity. So what is the importance of the parasitoid in the oil palm? So in this study, we use the parasitoid as a biological control to control the bedworm, because bedworm can cause a very serious infection. So about 33 to 40% of clot ulcers can be caused by that. So this slide, I would like to show that usually the healthy one of the oil palm leaf will look at the right side image there. So with the, at the left side there, and then the right side with the red arrow that is showing the infected oil palm leaf. So how the parasitoid usually kill the bedworms? So first they will mating first, and then they will always deposit the egg in the host, and then the egg will hatches in the host and eventually will kill the host. So that's why parasitoid can be served as a biological control as part of the RSPO criteria. And many studies show that the use of parasitoid actually can reduce the bedworm population. So why using the parasitoid to control the bedworm? Of course, first is no manpower required and also environmentally friendly and does not cause any human health issue. So usually in oil palm, we can find 3 types of the bedworm. So the first one will be the Medusa plana and Petroma pentula and Mahasana copperdii. So in this study, Medusa plana is going to focus because it is the most destructive bedworm to the oil palm plantation. So the targeted parasitoid in this study will be using the Badobius imperius because due to the limited study and longer lifespan and high fecundity. So there are 3 flowers of the bionomy that I would like to show. So the first one will be the Cassia copensis. Cassia copensis has been proved that it can favor by the Brachymeria species and also nematocysts, and it only just require minimum maintenance. And the second one will be the Antigona leptobus. It shows that it is a source of food or predators of the caterpillars, and it also very recommend for the oil palm plantation. And then the last one will be the Bidon pilosa. It is being known to attract the insect pollinator, and it is a very easy to grow plants. So in my study, the first objective will be to perform the Whitetail test on these flowers that attract the Badobius imperius females and analyze the volatile profiles of the flower that attract the Badobius imperius females using the SPNE and GCMS. So the research question will be, will the Badobius imperius female attract to this flower, and which chemical compound from the flower that can attract them inside the plants? So here are some of my overview methodology. So we're going to start the bedworm collection first, and then wait for the parasitoid to emerge from the pupa. And then the species identification was done by referring the handbook provided by MBOP. And these are the 3 parasitoid that was identified by using the handbook from MBOP. So, and then based on my objective, I'm going to run the Whitetail test to determine the attractiveness of the Badobius imperius towards these flowers, and then identification of the volatile organic compound by using the SPNE and GCMS. So this is how I set out the Whitetail test by using these 3 flowers. And then how I pairing the other source for the Whitetail test. So I'm going to run with blank first and then pair with each other. So for identification of volatile organic compound from flowers, by using the SPME to collect the volatile compound. So I'm going to absorb for 3 hours. Then after that, desorption in the GC injector port. Then this is the condition for the GCMS. So here's come to my result and discussion. So this is the general description of the Badobius imperius female. So you can see that this is the antenna, and then the compound eye, the forewing, hindwing, forelegs, mid legs, and hind legs. And then this slide, I would like to show the response of the Badobius imperius females in the Whitetail test. So the yellow highlighted one shows that it's a significant difference. And then the attractiveness of the flowers to the Badobius imperius female. So the Bidon pilosa is the most attractive to them, and then followed by the Cassia and then the Antigona. So here are some of the functional groups that can detect from this flower. So in the alkane group, we usually for the positive mean present and the negative mean absent. So in alkene, 2 sectant trimethyldodecanes, some of the studies says that this is the function of this is to attract to is very attractive to the Herosia vatisoimo, and the trigger is olfactory receptor neurons on the antenna. And then for the alkene group, we found that DMNT can as a cinnamon recruiting parasitoid foraging by predators and parasitoid, and also the carinophyllin serve as a feeding attractants and plays a vital role in giving a signal to rice white black plant stopper to locate their host. And then germacrine D can serve as a floral attractant and also for American cockroach attractants or for pheromones. And then it also can activate major types of olfactory receptor neurons on the antenna of parasite. And then come to the alcohol group. 1-hexadecanoate says that it's important during the mating process between male and female, and also had the function of enhancing the oviposition activities. And the methylsilandiol serve as an insect attractants, and 1-andecanoate increase the sex pheromone tract for the insect. And the last one, the ester, we found that the methylparaben says can increase the longevity and malaria oocyte abundance. So overall, we can say that why the Biden pilosa can attract the Pedobus impure females, because it has a high alkene group. So this group of scientists says that the alkene group have more intense odor compared to the other functional groups. And the other research also says that the alkenes can attract different pollinator species. So that's why the conclusions can say that this type of flower can as a food source to attract the Pedobus impure females. So Pedobus impure female were attracted to this flower, but however, it was strongly attracted to Biden pilosa. And then the GCMS analysis of the volatile extracted from these flowers has revealed some of the functional groups such as alkane, alkene ester, and alcohol might be the potential attractant to Pedobus impure female in selecting the plant. And then this is my acknowledgement, and thank you.
Thank you, Ms. Ong.
Yeah.
Thank you so much. All right. So if there's any question and any questions to her, you guys can still put it in the chat box. Okay, we will ask the questions later. Moving on next, I would like to call upon Ms. Nur Shabrina Muhammad Sharil. She's from University Malaysia Sabah. Okay. Ms. Nur, are you here?
Yes, I'm here.
Okay, you can start to share your slides.
Okay.
She's going to present her work on degradome sequencing reveals of miRNA 159 directed UH site in GAMYB, like gene during pineapple, ananas, commercials, fruit development. Are you ready?
Can you see my my slide?
I can't see.
Oh, okay.
Yeah.
Right there. Can you see myself now?
I still can't see. It's loading
Okay.
from my side. How about the rest? Okay, now I can see. Can you put in presentation mode?
Okay, it's a presentation mode now.
Yep, good to go. Let me start.
Okay. Bismillahirrahmanirrahim. Assalamualaikum and good afternoon to all respected professors, doctors, lecturers, and attendees. My name is Nushadina binti Muhammad Sharil from Biotechnology Research Institute, University Malaysia Sabah, under supervision Prof. Dr. Vijay Kumar and Dr. Nuhidayati Muhammad Yusuf. So today I'm going to present my study entitled Degradome Sequencing Reveals microRNA 159 Directed Cleavage Site in JMYB-like Gene During Pineapple Ananas Commerces Fruit Development. So microRNA in plant. MicroRNA is a small RNA of approximately 22 nucleotides that negatively regulate gene expression. In plant, more than 9,000 of microRNAs have been found in at least 83 plant species. And recently, the identifications of microRNAs and their target genes in diverse species have been a major focus of research. And these are some of the microRNAs and targets of and functions of the microRNA according to the species by the previous study, which these microRNAs were involved in regulate the plant growth, metabolism, and fruit ripening. In pineapple, at least 153 microRNAs have been found through in silico computational analysis. And these microRNAs play important roles in regulating the pineapple development. And microRNA 159 targets and silences the expressions of the JMYB-like family transcription factor, which play a crucial role in flower development as demonstrated in Arabidopsis and rice. However, in silico computational of analysis of the microRNA target's binding ability has only enabled the predictive identifications of genes targeted by microRNAs. The gene regulated by microRNAs will undergo degradation is yet to be proven empirically. And it is important to note that the main criterion used to distinguish between the genes regulated by microRNA is that targeted transcript will undergo degradation. And a way to do that is through the degradome sequencing. So degradome sequencing can be used to identify the microRNA cleavage site. There is advantage by using the degradome sequencing where it speeds up the experimental procedure in identify the functions of the microRNA as it does not involve the development of the transgenic line. And also it can be done on the large scale where the functions of all the microRNA in a specific sample can be done at the same time. And several studies have been done using the degradome sequencing to identify the microRNA targets and has been successfully applied to reveal the microRNA messenger RNA targets pair during the fruit developments of tomato, blueberry, grape, apple, and orange. MicroRNA works in a way that it will bind to the messenger RNA transcript. What happens after is the messenger RNA will be degraded. The difference between the messenger RNA that been degraded by microRNA is it's produced the pi monophosphate. For my study, the aim is to find the messenger RNA that been degraded by microRNA in pineapple, where first I will try to find the messenger RNA are expressed in the pineapple through the transcriptome sequencing, and second, I will try to find the messenger RNA that been degraded by microRNA with the pi monophosphate through the degradome sequencing. And finally, the alignments on the sequence obtained from the transcriptome, degradome, and also the microRNA will give me answer on what are the messenger RNA that been degraded by microRNA, and then what are the proteins these messenger RNA are coded for. So overview of the study. So the research question of this study is what is the role of the JMYB-like gene in the fruit development and ripening pathway, and how is the microRNA controls the expressions of the genes. And it is hypothesized that JMYB-like gene is a key gene responsible for pineapple fruit development and ripening, and is targeted by the microRNA machinery. We postulated that this can be empirically demonstrated through degradome sequencing. And the objective of this study is to identify the JMYB-like gene with the pi monophosphate that have been degraded by microRNAs in pineapple fruit through the development of the degradome library and bioinformatic analysis. So this is the full workflow for my project. For my samples, I collected the MD2 pineapple from Kota Blut, and for the extraction of total RNA, I was using a conventional STS method from Set et al. 2021. For the library preparations for both transcriptome and degradome sequencing, and the potential genes that have been degraded by microRNA was analyzed through the bioinformatic analysis and the validations of microRNA and microRNA targets expression by the RT-qPCR. And the last one is the verifications of genes that have been degraded by microRNA by 5'-res PCR. So for the cleavage site mapping of microRNA targets by 5'-res PCR, these are the modified workflow for the 5'-res PCR using the gene-res kit in vitrogen. The gene-res RNA oligo was ligated to the total RNA, and the PCR product was amplified using the gene-res 5' primer. and also the gene-specific reverse primer. After that, the final PCR product was gel purified and cloned into the CloneJack for sequencing. Comes to the result and discussion. GMYB-like genes identified in MD2 pineapple. So listed below is the microRNA 159 with the potential genes GMYB-like genes were found to be expressed in transcriptomic library and also regarded in the genome library, which also having a microRNA binding site and cleave at the required positions by the DSL1. RT-qPCR and data analysis. This amplification graph shows the CT value of reactions of the amplifications of reference genes beta-actin, microRNA 159, and also the microRNA targets. According to the melting curve graph, there was no unspecific product detected. And the graph here was plotted according to average relative expression value of the 3 replicates of microRNAs and also the target genes. And for the messenger RNA amplification by Pi5' PCR, the listed is the primer that I have designed and used in this study. And the figure 8 shows the amplifications of the target genes on the 3 biological replicates of MD2 pineapple through the Pi5' PCR, and figure 9 shows the gel photo of the extractions of the plasmid DNA containing the microRNA targets. So this is the chromatograph of the GMYB targets from the single sequencing, and from the sequence, this is the gene reser RNA oligo that been ligated to the RNA, and this is the cleavage sites of the GMYB-like genes, which is being targeted by the microRNA 159. And the black arrow here is indicates a cleavage site, which is verified by the Pi5' PCR. And this is the T-plots that generated based on the duodenum take of the microRNA on the transcript, and the T-plots here shows the GMYB-like gene was cleaved by the microRNA 159 at the positions of 1, 2, 4, 6 of the transcript. And this cleavage site is the point of the degradations on the transcript, potential target genes by the microRNA, which is at the positions of 11 nucleotides of the microRNA. So as the conclusion, GMYB-like transcript mapped to microRNA 159 were found to be consistently expressed in all 3 biological replicates on both transcriptomic and degradome libraries, thus speculated to be the true targets of the microRNAs in pineapple. And conversely, the GMYB-like genes is negatively regulated by microRNA 159, were found to be involved in regulating the plant development, and the target messenger RNA were detected by RT-qPCR in MD2 pineapple. A modified 5' PCR was performed to validate the gene targets of the microRNA 159, and these findings confirm that GMYB-like family transcription factor is an authentic target of the microRNA 159 and demonstrated the key roles of the microRNA in regulating the expressions of target genes, providing insights into the mechanism underlying the MD2 pineapple fruit development. So this is some of the references that I've been using in this presentation. And before I end my presentation, I would like to acknowledge the research fund of this study, FRGS, and also Scheme MS Grad, and also to Genomic Laboratory members for helping me throughout this study. Thank you.
Okay, thank you so much, Ms. Nur Shadrina. Okay, so wonderful presentation. Okay, let us move on next to our last presenter for this session. Let me call upon Mr. Muhammad Naim Rosli from Management and Science University, Malaysia.
Hi.
Yeah. Can you start with your slides?
Sure. I will share my presentations.
Okay, Mr. Naim Rosli is going to present on the effect of feeding probiotics, phytobiotic, and prophytobiotic on egg quality, total lipid content, and family acid composition of egg yolks of laying hens. All right. Now it's lunch hours. Okay, we are talking about eggs and chicken.
Yeah.
Yeah, I can see your slides and you can start now.
All right. Thank you to the chairperson. Assalamualaikum and very good afternoon to all of you. Yeah, currently is a lunchtime, so I would like to present about the effects of feedings probiotic, phytobiotic, and prophytobiotic, actually a combinations of probiotic and also phytobiotics on the egg quality, total lipid content, and also fatty acid compositions of the egg yolks of laying hens. So this is our product by MSU itself. So why actually this slide is not moving. Okay. So why actually I choose these probiotics? Actually, probiotic, phytobiotics, and other natural products provided a gut ecological immunities, antioxidative status, and also improve the qualities of laying hens itself. Actually, is a combinations is considered as antibiotic growth promoter for poultries. There's an urgent need currently in agriculture industries to develop new item to replace the antibiotics for food producing animal, particularly in poultries and also livestock. So if you can see from the graph here, since 2016 until 2021, we can see that we are actually a largest eater of eggs, from 20 to 22.8 kilogram per year. We also a large capital egg consumptions across the world. So what actually I'm using Backhouse Cichadora, we call as lemon myrtle, currently native in Australia, but we already actually we brings from Australia to Malaysia due to their unique flavor, fragrance, and also biological properties. It's currently been planted in Sekinchan, Kluang, Kuala Linggi, Sabah, and also Hulu Langat. It's already past the Australian standards, and currently this plant actually natural, less toxic, and also free from residue. What actually problem that we are facing now? Actually, WHO has called up to stop and also minimize the use of antibiotics to prevent the spreading of antibiotic resistance. These accumulations of antibiotic resistant and poultry meats and eggs will increase the development of multi-drug resistant bacteria, which currently is very, very hard to treat in humans. So they also have a controversial issue on synthetic antioxidants such as BHA and also BHT, whereby this help to slow down the lipid peroxidations in poultry productions, which currently, if you are eating too much of it, will lead to cancer. They have limited studies in utilizing essential oil in poultry industry, which is lemon myrtle, currently limited to pharmaceuticals and also healthcare industry in Malaysia. So what is the objective of my study is to evaluate the effects of feeding probiotic, phytobiotic, and prophytobiotics on egg quality, total lipid content, and fatty acid compositions of laying hens. This is my overall methodology, whereby I collected the lemon myrtle leaf from plantation area in Kuala Linggi. Then we are doing lemon myrtle extractions by doing hydrodistillation method. Then I'll check the quality, total lipid content, and for fatty acid compositions, whereby each of 4 weeks from 20 to 36 weeks of age for the hens itself. So what I'm looking for egg quality, I'm looking for shell thickness, egg specific gravity, albumin and yolk egg weight, Hox unit, and also yolk color. For total lipid content, this one I'm using chloroform methanol methods. And for fatty acid composition itself, this one we are using direct methylations method, and we're analyzing by using gas chromatography. So, one moment, yeah. So in vivo studies, we are using Lohmann Brown's hens, which at the age of 20 weeks of age, randomly allocated to 4 dietary treatments. So my first groups, which is control diet, we only giving them with basal diet. The second one, which is probiotic, combination of basal diet with B. amylase quercetin CECT 5940. The next one, the third one, which is phytobiotic, combination of basal diet with 1 ml of lemon myrtle per kilogram. And the last one, which is combination for both, which is the probiotic and also phytobiotics. So let me see on the result itself. If you can see from this table, there is actually no significant figures towards the specific gravity, shell weight, albumin weight, yolk weight, shell thickness, Hox unit, and yolk color. These have been proved by a few other researchers when they are using Aviva oil and also oregano essential oil, whereby there is no significant albumin height, Hox unit, and also egg yolk color. And for oregano oil, there is no significant on shell weight, shell thickness itself. For B amylase, which is probiotics, there have two probiotics that I'm looking for, which is no significance on albumin height and also yolk color. And for B subtilis and B liverniformis, also showing no significance on the egg weight, albumin height, yolk color, Hox unit, and egg shell thickness. What these findings have presented and previous study were that supplemented with probiotic phytobiotics and also probiotics actually did not give any effects towards the Hox unit and proportions to albumin, yolk colors, specific gravity, shell thickness, and also there is no negative impacts towards the egg quality itself. Let me see on the total lipid contents from week 23 till week 35, also showing there is no significance figures. What is this indications actually from previous studies showing there is no actually significance towards a total lipid content when they're providing with probiotic itself, but they have ones which is currently had been shown when they are providing with Akramanaceae muciniphila, which is one probiotics, showing they have a significance figures on the total lipid content itself. So when they are filling with probiotics, which is flaxseed oil and also thymus, saga, and rosemary oil, showing there is no significant figures showing on the total lipid content. Why this is happening? Actually, observations by Kalavati and others showing significant reductions in abdominal fats depositions and serum lipid in the boiler chicken, supplemented with the same lactobacillus-based probiotic used in laying hens 2019, showing there is no significance. The lipid protein metabolisms of laying hens varies from the immature hens and male birds, which might to some extent explain the variety efficiencies of probiotic, phytobiotic, and probiotic in lowering down the lipid content of the boiler chickens' eggs in laying hens. So let's see on the last one, which is fatty acids composition, which is the main one. If you can see on the week 23, showing there is a significant figure showing on the total unsaturated fatty acids. And then they have a also significant in total polysaturated fatty acids, omega-3, omega-6, and omega-9. Omega-3 and omega-6, this one is very essential fatty acid, which is unable to produce by our body itself. So we have to put an intake towards it. And for total polysaturated and unsaturated and also monosaturated, we consider as a good fatty acids, whereby total saturated, this one we consider as bad fatty acids. So if you can see on the next slide, on week 27, showing they have an improvement a little bit on the total saturated, monosaturated, but there is no significant figures on total polysaturated, but the omega-3 and omega-9 remain, which is have an improvement. The next slide showing on week 31, showing for all they have actually significant figures. For the last week, which is on week 35, showing there is only don't have any figures, significant figures on omega-6. So what this indicate, if you can see from the slide itself, they have a three probiotic have been showing there is no reductions on the total saturated and increased unsaturated polysaturated fatty acids. The next one, which is lactobacillus-based probiotic, showing they have a reduction, the total saturated, and increase the unsaturated fatty acids. On the next one, which is lactobacillus paracillis, L. ramonosus, and L. ramonosus, which is this is one of the combinations, help to reduce the saturated and increase the unsaturated, also monosaturated and polysaturated fatty acids. If you can see on the part of phytobiotics itself, oregano essential oil, improve the monosaturated fatty acids and improve omega-3 fatty acids. By bergamot essential oil, this one when we are they are feeding with it, is improve the polysaturated fatty acid, improve omega-3 fatty acid as well. Why these things happen? That the profile of fatty acid in egg yolk strongly depends on the food diet that the bird digestive system and also biosynthetic process of the laying hen itself due to the change in various enzymatic process in the yolk sac membrane and change in lipoprotein transportations. And also there have several studies have postulates that the change in fatty acid profile of the laying hens by essential or supplemented laying hens were related to the change in lipid metabolism, but is in need for investigations. If you can see from the results slide, I'm not showing any potential for combinations because there is none. There is no results towards the combination of probiotic and phytobiotics. But our product has shown there have a significant figures when we are comparing it towards control. So what actually the conclusions I'm putting on this project is that the finding indicate that supplemented of probiotic, phytobiotic and prophytobiotics in the hen's diet was able to improve the fatty acid compositions of egg without affecting the egg quality and also the lipid total lipid content itself. So this is my reference, and thanks to MOH for funded this project and also my research team, including my collaborators from a few industries. And thank you. If you can explain it simple, you don't understand it well. Thank you. Over to you, Chairperson.
Right. Thank you so much, Mr. Muhammad Naim. Right. Very nice and interesting presentation. Okay, that's the end of our oral presentation session for today. Okay, but we still have Q&A to go on, right? So I've received few questions, okay, from the floor. So we can kickstart with that. I hope all the speakers are still here. Okay, we shall start back with the first speaker, Dr. Ikhwan. Dr. Ikhwan, are you here?
Yes.
All right. So how are you doing?
I'm good. Thank you.
Okay. So regarding your project in terms of omics, okay. So this multi-omics approach is quite recent, right? So and how you think that this method is beneficial in terms of understanding the Ganoderma body science compared to other classical methods?
Okay. Right. Thank you, Dr. Bhaskaran, for the question. So basically, as we know that the conventional genetics, so they are doing the, it takes time. So actually, when we compare to the modern technologies that we have, so in terms of creating or getting the results, it's much faster if we are using the modern technologies. And actually for that, moreover, in terms of the knowledge that been gathered from the multi-omics data, so we can improve in terms of the development of diagnostics, or in terms of the development of how to say that, to combat the disease caused by the Ganoderma boninens. So basically, actually, the presentation that I did just today is just a very tiny bit of the project that we are currently doing in UKM. So we are trying to focusing on in terms of redefining what is the important genes that are responsible for the infections of the Ganoderma boninens. So from that, we hope that we can try to design the improved methods of the detection methods based on the genomics data and transcriptomic data that we have. So I think that's the answer for your question.
All right. Thank you so much. I hope the project will keep on going and, you know, do give significant impact on our oil palm industry. Thank you so much, Dr. Ikhwan.
All right, thank you, Dr. Bhaskaran.
All right, moving on to Miss Siti Dalila. Right. She started to smile already. Okay, so this MATAC project, may I know why it is being the interest under ICMBB plantation? And is there any other groups, agriculture group like RISDA or even we have Jabatan Pertanian Malaysia are doing similar projects to this?
Okay, so in regards of mata planting material, actually it has been introduced by Jabatan Pertanian (DOA) since many years ago, and there are other plantation industry like United Plantation that actually involved in supplying high number of mata for the planters in Malaysia. But in regards of the markers application, so far we learned that we are the first one that actually applied this kind of marker screening to basically watch or monitor our mata purity level. So it is not easy actually to get the real mata because we have only the seed producer which who have accessibility to the parental pumps that can do it. So there's only a few in the country, and those that don't have the parental pumps, they tend to sell the F2 generation of mata, which is not exactly have the characteristic that true mata have. So it is important to actually identify the real mata to sell to the planters. So this is one of our way lah to do it.
All right. So I believe there are many matak, species that is being sold outside there, even for the farmers. So the issue now is whether we don't know whether it's a real matak or not.
Yes.
That is one of the issues, right? So in terms of matak, why matak is being considered as one of the premium coconut, compared to other hybrids?
Actually, there are many hybrids that being between the that come from the cross of the dwarf type and tall type of coconut. There are several hybrids that being introduced by Jabatan Pertanian, but mata have been studied that have the most superior basically in terms of yield, the number of fruits that it produces is like a higher number, and also the size of fruit is larger because it follows the tall type punya fruit size, but the number of fruit it follows the dwarf type, so it get like best of both characteristic of the coconut type. So this is the specialties lah. This is the superiority, and it's versatile, like as I shared in my presentation.
Right. Okay. Thank you, Ms. Siti.
Thank you.
Right. If you guys still have any other questions to her or our earlier presenters, kindly put it in the chat box. Okay. Moving on next, Mr. Aminu. Okay. So, Mr. Aminu, in terms of the soil sample collection. So may I know where you have done your sampling, and can you explain on that part again? Can you unmute yourself?
Yeah, can you hear me?
Hello, can you hear me?
Now I can hear you.
Okay. For the sampling, for the arsenic rich, we try to go to the Rahmantin hydraulic in Perak for the arsenic rich, which is an extinct mining, and so we collected the sample from there and analyzed the arsenic, the heavy metal content, arsenic content, and then arsenic speciation in the soil. Then the natural mineral soils samples were taken from within the University of Science Malaysia campus.
Right. So may I know why specifically certain areas you are taking from the industrial areas? Is there any other previous studies that leads your sampling towards that area? Or
yeah, there was a previous study that actually carried out research on that heavy on lead and cadmium content in the peroxide specifically. So it was, but in different plants, Mimosa pudica. So it was observed that there is a high amount of cadmium and lead in the area. So when it comes to my own research, I'm using Teres pitata. So I try to see whether the Teres pitata also can be found in that particular location. And if they are available, then is there any relationship between the other heavy metals content in the area and the Teres pitata accumulation? Then I try to compare that with the non-contaminated or naturally, or let's say natural mineral soil, because from the result, it shows that it is not contaminated because the amount of heavy metals, arsenic specifically, there is around 6 to 5 milligram per kilogram, compared to the RHT, which has around 101 to 3,161 milligram per kilogram arsenic, indicating that the amount is too high in the contaminated area. So based on that, we try to see, and then this is just part of the, let's say part of my PhD work. So we have carried out so many work on these two soils. Bacterial isolation was carried out where we isolated around 124 microorganisms, which were all proven to be plant growth promoting microorganism bacteria and also arsenic tolerant. So we try to subject those microorganisms to the growth of the plant and see how they can improve on the hyperaccumulation of these heavy metals. Yeah.
So in that case, can I say that the level, the concentration that you got through your studies is above the maximum permissible limit by Department of Environment?
Yes, it's above the maximum limit in the arsenic-rich area.
All right, very interesting. Okay, thank you so much, Mr. Aminu.
Okay, thank you very much.
Okay, moving on next, we have Ms. Ong Chin Yin. Okay, so in terms for you, okay, there's a question on, can you summarize the outcome of your research in terms of the application in the agriculture industry or oil palm related industry?
Hello.
Yeah. Can you hear me?
Yeah, can repeat the question again? Sorry.
Yeah, can, no problem. That's my job today as a chair. Okay. Can you summarize? Can you summarize the outcome of your research in terms of the application? How are we going to apply your research in the industry, agriculture industry?
Okay, because all right, in my study, like by understanding the specific compounds in the flower, they're able to attract the parasitoid. Maybe we can develop the targeted strategy, like for example, like develop the synthetic attractants that mimic the natural flora signals. And then by doing this way, we can enhance the efficiency of the parasitoid as a biological control agents. And then that means that by doing this way, we can provide more environmentally friendly alternative to the chemical pesticides and reducing the pesticide usage. And then also can help the farmers to reduce the reliance on the synthetic chemicals where still effectively managing the badworm infestation.
All right. Okay. So in terms of your study, have you done it at your university or you have any other station place for you to?
So far for my study, I done the YQ test in my university, and so far the other study also got done before, but they're just based on the observation. So they never used the method that I set down just now, showing the slide of YQ test. So the YQ test, the purpose of using the YQ test is just we want to know that the preference of the parasitoid to which flower. So this is the difference from my study compared to the other researchers.
All right. All right. Thank you.
Thank you.
Okay. Moving on, Miss Sabrina.
Yes, I'm here, doctor.
Okay, so in you talk much things on pineapple development, happening, this kind of process. You have looked into one gene, okay, GAMYB, if I'm not mistaken. You have looked into that particularly, and why you don't look into other genes? Why specifically this one gene?
Because this actually it's already been studied by previous study, but not many of the species they are actually focusing on this gene. So for specifically for this my study, I just focused on this gene to add more informations on this gene which is involved in the regulating of the plant developments of the pineapple.
That means, can I say that people have done a similar study using the similar gene on pineapple?
Yes.
Then what's the difference of your study compared to that?
So the difference is for this particular for my study is to focus on this pineapple, but for the other study is focused to other species. So for this my study is focused to the non-climatic fruit itself for the for pineapple. Yeah.
I see. Yes. Yeah. All right. So what are the difficulties that you found when it comes to your project, you know, especially because it's not that
the
easy.
difficulty I think it's more to the lab work for my farm PCR, where I validate the target gene, that because we want to capture the one that are really been cleaved by the microRNA, so we have to specifically design the primer, which is really the specific primer that we can capture the cleave site of the microRNA. So we have to study the sequence of the transcript more, so we can understand how, and then we can design a good primer for the performance PCR. I think that is the difficulty of this study lah.
Right. All right. Thank you. Wish you all the best. Right.
Thank you, Doctor.
Yeah, moving on to our last presenter. So, Mr. Muhammad Naim.
Yes.
So in this case, yeah, I found that you are working a lot with biotics, okay, probiotics, phytobiotics, phytobiotics. Wow, very difficult. So in this case, antibiotics is something that people are working in a very easy manner right now. Yeah.
Correct.
So one of the major reasons for that is definitely cost is coming into the picture, right, when it comes to the industry. So now you are coming out with something that is quite promising, quite new to the industry, but how, when it comes in terms of cost perspective?
For the cost itself, I do apologize, Prof itself, from my end here. I am unable to answer because this is still under studies, still under investigation and also studies. So I believe inside here there's a lot of industrial people. So I hold these questions until the products released to the market itself.
All right. That means you have to do more literature review on that part as well, because you are developing things from the scratch. Means this cost consideration should be there if you are looking into the industrial application. Anyhow,
Yes.
when we are looking into this study, okay, let's say we are comparing your study with the antibiotics, okay, straight away. So what are the major difference that you can see, the significance out of your project?
The significance difference between probiotics and also phytobiotics, along with probiotics, you are comparing, if you are comparing with antibiotics itself, if you can see antibiotics develops a lot of causes when it comes to human interactions, meaning if you're taking much of antibiotics, if you can see from my problem statement also stated that this will lead to developments of antibiotic resistance, whereby it's very, very difficult to the persons from the healthcare industry itself to manage these situations. It's not only coming from the medical line itself, from industry itself. When it comes to antibiotic resistance, it's very difficult to treat other disease in the animals liabilities, I mean, in the animals sites itself. So what we are looking for, we are trying to actually improvise methods, or we call as, we are trying to replace the use of antibiotics with other one, which is prevention's better than cure. So we are promoting this. We are prevent it before it can go worse. So if you can see from previously, we have a lots of issues with poultry such as H1N1. So this causes a lot of loss towards the industry itself, and along not only industry impacted, us as humans also impacted with this. So by preventing it, we can also preventing other possible measures or other possible disease that can occur towards us itself, Doctor.
All right. Okay. Good. So yeah, prevention is better than cure. Okay. It's not only applied for humans but also for animals, right?
Yeah.
Okay. Good. So I think yeah, that's all for you. So thank you very much for your presentations, and thank you everyone for your kind sharing. Okay, if you guys have anything to ask me, you can ask me now. All right. So before we end this session, okay, I would like to thank you for all of you to follow the time just now. I think personally, I do learn a lot from your projects. It's a variety of projects, and so many things to learn. Science is diverse, right now. So just following our theme for today's conference, everything is transdisciplinary. Many things can be learned at the same time. Okay. So just to other participants here, so we have shared the your attendance link. Kindly click in that, put in your attendance. Okay. By doing that, only you are going to get your certificate and so on, right? Okay. If there's no other questions, let us resume this session today at 2 o'clock later. This 1 hour is for your lunch break. Okay. So thank you and have a nice day, everyone. See you. Bye. See you later, 2 o'clock.
Hello, good afternoon. All of you, can you all hear me from the other side?
Yes.
Oh,
Yes.
thank you for the response. Thank you. So let me check on the sound bell here. Can you hear the bell?
Yes, can, but
Okay.
louder will be better.
Louder. Okay. This is the max I can put already, so I will put it closer to my mic then. Okay. Okay, great. Okay. So now we have our concurrent session number 3, the third concurrent session of the day today in medical forensic biotechnology and molecular biology. Okay. So this is our oral presentations. I am Dr. Choi from UCSI University, Malaysia. Okay, nice to see all of you here. I think today we have a big crowd. So I hope everyone has your lunch done. Yeah, okay. So now we are here again, back again for our third sessions. We have our first oral presenter for these sessions, O15. Okay, Mr. Ziadin Azimi.
Yes.
Okay, you
Good
may try
afternoon.
to. Good afternoon. So you may share your slide now.
Yeah, sure.
Yeah, we can see your slides. Can you put it up for full screen? Okay, great. Okay, so before Mr. Ziadin start his presentations, okay, we have several housekeeping announcements here. There are our technical team have posted some of the housekeeping announcement at the chat box. Okay, please to remind everyone to put in your attendance. Okay, and with that we have Mr. Ziadin Azimi to share with us his presentations on micronutrient deficiency and its association with ADHD and academic performance of primary public school students in Kabul city, Afghanistan. So Mr. Aziuddin, you have 10 minutes for your presentation. So I will ring my bells at 9 minutes once and up to 10 minutes, that's twice ringing. Okay?
Okay.
Okay, you may start your presentation. Thank you.
Okay, thank you. And good afternoon to you. My name is Ziadin Azimi, the title as you read. For introduction, many studies have been linked the food consumption or type of food with attention effect hyperactivity disorder, and also the academic performance, but correlation or association of micronutrient with this disease of this form or academic performances unknown. As we know, in many countries, micronutrient deficiency is common, especially in developing countries. But unfortunately, in Afghanistan, we don't have studies to show the prevalence of vitamin A, D, B12, folic acid, calcium, and zinc deficiency among primary school children or school age children. The objective of this study was to find or to study the relationship between nutrient level and academic performance of primary public school children in Kabul city, just in Afghanistan. The specific objective was to determine the prevalence of micronutrient deficiency among this study population, first. Second was to perform the psychometric property of 2 questionnaires, one for ADHD, and the second was academic performance among this study population. And the third one was to determine the association of micronutrient deficiency with ADHD and also with academic performance in this study population. To do this study, among 50 schools were present in 14 districts in Kabul city. 3 districts was selected using simple random sampling, District 3, District 5, and District 11. In these districts, 5 schools was again selected using simple random sampling. Schools with primary school children, girls and boys aged around 7.7 years. 511 students were recruited in this study. Among them, 358 was included and 153 was excluded from the study. For three objectives, firstly, micronutrient level for vitamin A, D, B12, and folic acid was measured using ELISA kits, and the calcium and zinc was measured in their plasma by colorimetric methods. The two questionnaires, ADHD teacher vision and academic performance questionnaire, both was translated, validated for the psychometric preparatory, and the association of this micronutrient level among the children with academic performance and ADHD was examined. For the question validations and translation, first the translation of ADHD and academic performance, both questionnaire was translated using standard way for the backward translations. The content quality was tested by a panel of experts from 3 departments of psychology, constellation, and pedagogy departments of the Faculty of Psychological Science. The face validity was tested by well-experienced teachers. There were a number of 40 teachers. And for the concept validity, confirmatory factor analysis for both questionnaire and confirmatory factor analysis only for ADHD was measured for 1, 2, and 3 dimensional component. Reliability was tested using the Cronbach alpha, and finally the test was the questionnaire was rated by the teachers, as you can see the pictures, by 40 teachers for each of their students. The result for the prevalence of micronutrient deficiency showed a very high prevalence of vitamin D, B12, and calcium deficiency among these children. Also, the prevalence of vitamin A and vitamin D deficiency was also present among this population. And also there was a nearly lower, we can say, of nearly 5% of the population of the study was folic acid deficient. For the objective 2, the content validity showed for both questionnaire very good interlevel and item level content validity and scale level content validity, and also the kappa value, which showed excellent, and it was acceptable for the ADHD and also for the academic performance questionnaires. For both of them, the face validity result also showed similarly to content validity. It was very excellent with item level face validity index and scale level validity, and also the kappa value. For both questionnaire, ADHD teacher vision, which was translated to Dari is one of the language in Afghanistan, we speak here, especially in Kabul, and also for the APQ. And the concept validity showed for the ADHD to the two-factor loading, which was very near the correlation of text was very near to 0. The Boret test was also acceptable, and the KMO for both questionnaire was higher than the level which was recommended 0.5, but for the test what it showed 0.913, and also for the APQ which also was very high, 0.935, which was acceptable. The confirmatory factor analysis for the ADHD, we use 3 tests, 3 factors loading, one for 1 factor loading, as we can see, and also for 2 factor and 3 factor as well recommended by the literature and other studies. Our model showed best fit for 2 factors, as the original questionnaire was also the same for the 2 factors. But some studies showed 1 and 3 was examined that. The reliability for both questionnaire was very acceptable. Reliability for the 2 scale of ADHD was higher than the recommended 0.75 threshold volume, and also for the total ADHD. For the APQ, it showed also very good result for Cronbach alpha, and it was acceptable. The result of the ADHD among the students which we examined, we can see the figure. The female student put lower inattention and also hyperactivity passivity, and also the total ADHD compared to the male student, which was subsequently very significant, lower than the male students. About the correlation of the micronutrient level with ADHD, we only find that vitamin D deficiency showed significant negative correlation with inattention, and vitamin B12 deficiency showed significant negative correlation with hyperactivity and also with total ADHD, but not the other micronutrient, and it should no significant correlation with ADHD or their subdomains. The academic performance showed female students have higher score for the academic performance compared to the male, but statistically it was not significant. Correlation of micronutrient with academic performance, we cannot find any correlation, significant correlation of vitamin A, D, B12, folic acid, zinc, and calcium with academic performance in this study. Correlation of ADHD with academic performance, as you can see the figure, the table here. Inattention, hyperactivity, and impulsivity, and ADHD. Three of them, or we can say two domain and total ADHD, had significant negative correlation with academic performance. And also this test was confirmed by the regression analysis. You can see the regression. The inattention had 12.6%, hyperactivity 8.3%, and ADHD 12.8% impacted the academic performance among both girl and boy, especially the boy was more significant. In conclusion, we can see high percentage of students had vitamin D, vitamin B12, calcium deficiency, and also there was vitamin A and folic acid deficiency with zinc deficiency as well. The translated quotient of ADHD and APQ should good for psychomotor property, and we can be used in future in other studies for the testing the ADHD and also for the academic performance of students. And negative correlation of vitamin D and will only we find to have negative correlation with ADHD or with its subdomain. No significant correlation was found between academic performance and micronutrient level. Significant negative correlation was also found between ADHD and APQ and its subscale of ADHD. And finally, now I want to acknowledge many people, especially the student of Faculty of Pharmacy. They helped me, the parent, the student. I repeated the study many times because of some problem here. They all helped me during the study, and I want to thank all of them, and also the donor, and which they supported or granted our study. Thanks. So it was all from me.
Okay, thank you, Mr. Ziadin. I think this is a very interesting topic. Okay, so let's us move on to our next presenter. Just to remind everyone, you are free. Please feel free to drop your questions, okay, or through the chat box, and we will have our Q&A sessions. We will have our Q&A sessions at the end of the entire concurrent session 3. Okay, so we have our next presenter, okay, Miss Kee Lee Teng. Okay, Miss Kee is from University Kebangsaan, Malaysia. And Miss Kee, please share your slides, share your screen.
Sorry, can you see my screen now?
Not yet. No. Can you try again?
Oh, now also
yes.
no?
Yeah, now it's okay. Okay, put up screen.
Okay. Hello?
Coming. No, I can't see your screen. Can you try again? Just now it was okay, but we cannot see a full screen. But now it's totally no sharing.
Hello.
Yes.
Can you see my screen now?
No. No.
Okay.
Yeah. Now it's full screen. Yeah.
Okay.
Good.
It's in
Okay.
presentation mode, right?
No. This is full screen mode. This is full screen mode. So Ms. Key, okay, you have 10 minutes for your presentations, and Ms. Key is here to share with us his studies, his research, her research in evaluate the potential of fibrinogen depleted human platelet lysate for mesenchymal stromal cell expansion. The floor is yours, Ms. Key.
Okay, very good evening to everyone. My name is Ki Li. I'm from Center for Tissue Engineering Center and Regenerative Medicine. Today, I'm going to present my research with the topic of evaluate the potential of fibrinogen depleted human platelet lysate for mesenchymal stromal cell expansion. As we know, FBS is a common cereal supplement for in vitro cell expansion. However, it is not ideal for clinical use due to the risk of animal pathogen transmissions and transplanted hosts. So, it's not due to this disadvantage, it is not compliant with the GMP. Therefore, human platelet lysate is proposed as a replacement for FBS for in vitro cell count growth. Human platelet concentrate is a expired platelet concentrate that contains a variety of growth factor needed to maintain the viability and growth of culture cell. It has a few advantages of using HPL, which is no active blood withdrawal, since the expired platelet concentrate is collected and there is low transfusions and transmission infection collected is from low-risk populations and screened. Besides, they are genogenics free. In our previous research, we have found that in-house HPL promoted more effective proliferation than FBS in different cell type, which is fibroblast, chondrocytes, and umbilical cord mesenchymal stromal cell. However, for the HPL, they contains fibrinogens and other coagulation factor, which induce gel formations during cell culture. To prevent these situations, heparin is frequently added. However, for the heparin, it is primary and it can only reduce the chance of gel formations. Moreover, high concentrations of heparins linked to the suppressed MSC expansions and also the outer cell morphopose and alternative approach for the fibrinogen depleted HPL preparations and for the expansions of mesenchymal stromal cell without heparin. My research objective is to prepare the fibrinogen depleted HPL that support heparin-free expansions of mesenchymal stromal cell. In this study, we will collect the expired platelet concentrate were lysed using mechanical lysis to obtain the HPL. Then the host HPL were divided into two portions. One of the portions were added with the heparin and classified as the HPL. Another portions will undergo the fibrinogen depleted process using calcium salt and then classified as FDHPL. Both group of the HPLs were quantified with their growth factor, calcium concentrations, and fibrinogen concentrations. So umbilical cord mesenchymal stroma cells were cultured with 10% HBl, FDHBl, and FPS to determine their cell viability, population diversity, cell morphology, and total cell number. And the UCMSC that culture with FDHBl were characterized for their immunophenotype and multilineage differentiation potential. Next is my result discussions. From the figure, we can see that in HA is unclear and the precipitate formations in the FDHBl that is clear and no precipitate form, which indicate from during the fibrinogen process, depleter process. Next, we also determine the calcium concentrations and fibrinogen concentrations in HHBl and FDHBl. From the results, we can see that the calcium concentration in FDHBl is significantly increased compared to HHBl. This is because calcium salt is added during the fibrinogen depleter process. While for the fibrinogen concentrations, it has a HBl when compared to HHBl. Although it's a mild reductions in FDHBl, it is low enough to avoid added the heparin to prevent the gel formations and the precipitate formation during the cell culture. Next, we also quantified the growth factor in HHBl and FDHBl because we worry that the growth factor will deplete during the fibrinogen depleter process, and we determine the 4 growth factor, which is FBD and F. Because these 4 growth factor is important. This growth factor are very important in promoting self-growth and maintain cell survival. From the result, we can see that there is no significant difference in BDNF, TGF-beta1s, and PDGF-BB, only in a significant reductions in VEGF. This is because we think that VEGF have bind to the fibrinogens and fibrins with very high affinity. Then we also determine the morphology of UCMSCs cultured within different cereal supplements. And all the groups demonstrate a similar long-spindled fibroblastic morphology, while there is no significant difference observed in HHBl and FDHBl groups. Then we can see that in HHBl groups and FDHBl groups, they are smaller in size when compared to FPS group. Next, we also characterize the UCMSC, the culture with FDHBl. From the result, we can see that UCMSC, the culture with FDHBl, express the MSC specific markers, which is CD73, CD90, and CD105. They are negative for the hematopoietic markers, which are CD34, CD45, CD11B, CD14, CD19, and HLA-DR. Next, we also characterize the UCMSC for the multi-lineage differentiation, which indicate that the MSC able to differentiate into adipocyte, while for the osteogenesis differentiations, there is calcium deposit formations during the osteogenesis differentiation, which indicate that the MSC able to differentiate into. Lastly, we also determine the cell viability and population doubling total cell number of UCMSC with different cereal supplement. From the results, we can see that there is no significant difference between the cell between HBl and the FDHBl in the cell viability and population doubling. They shows the significant difference for when compared to FPS, and there is no significant difference among all the group. However, it shows the mild reductions in FDHBl when compared to total cell number. This might due to the amount reductions of BDGF, then that is very important for the promoting self growth and maintain the cell survival. In all these results suggest that the high concentrations of calcium and the reductions of VEGF did not affect and growth of the MSC, since there is no significant difference in cell viability, total cell number, population doubling time, and also multi-lineage differentiation potential and the self-surface marker. So in conclusions, FDHPL enhanced in vitro expansions of MSC while maintaining their cell characteristic intact, indicating its potential as a substitute for FBS in genogenic-free MSC expansions. This study was financially supported by the Faculty of Medicine, UKM. And this is my reference. That's all from me. Thank you.
Thank you, Ms. Key. Just on time, on the dot. Very good. Thank you, Ms. Key. Okay, so let's have our third presenter, 017. Okay, Ms. Pavitra, are you here with us?
Yes.
Okay, you may want to share your screen. Ms. Key, please unshare your screen. So Ms. Pavitra Nanda Gopal is from University Technology Malaysia. I can't see your screen. Are you sharing? Wait. No, I can't see your screen.
It's loading.
Okay, maybe coming.
Yeah.
No, it just says started screen sharing, but we can't see anything.
Oh God. Yeah.
Can you try again?
Okay, wait on.
Yes. Coming is here. Okay, I can see your screen now. Okay, so try to put on a full screen.
Full screen on. Okay. All right. Okay.
Okay, all good. Okay. So yeah, we have Miss Pavitra here with us. She's here to share with us cloning of plasmid for compatible gene expression in cyanobacteria. Again, you have 10 minutes. The bell will ring at 9 minutes and at the end of 10 minutes. Okay? The floor is yours, Miss Pavitra.
Thank you, Professor Dr. Choi. A very good afternoon to everyone. I'm Pavitra Nandagopal from UTM. I would like to present a part of my PhD project entitled Cloning of Plasmid for Compatible Gene Expression in Cyanobacteria. So first, what is cyanobacteria? Cyanobacteria are known as blue-green algae, which have been used to study photosynthesis process and its structure. And why cyanobacteria has chosen as a emerging host for metabolic engineering research. It's because cyanobacteria has the ability to capture energy from sunlight and converts carbon dioxide to range of biofuels like alkanes, terpenes, and also fatty acids. And there are two genetic modifications for cyanobacteria. One is INSYS, another one is intrans. So in this study, I've been using INSYS because the plasmid that I have, the genes that I have cloned into a plasmid has neutral site. And the research gap of this project is there is no expression plasmid used for production of flavonoids in thermophilic cyanobacteria. And the objective of this project is to clone a plasmid carrying flavanone biosynthetic genes for a compatible gene expression in Cynococcus elongatus. And here are the genes that I have used for cloning. So the first one is CHS. CHS was synthesized by Petunia hybrida with the size of 1,170 base pairs, whereas CHI was produced by Pureria lobata with the size of 745 base pairs. So these both genes actually were synthesized by GenScript. And here comes the seamless cloning. You must be wondering how I clone the genes into a plasmid. So seamless cloning was used in this study. It's a two-step process consisting of in vitro assembly followed by transformation. The enzyme mix uses proprietary enzyme mix to assemble DNA fragments without extra sequence. So the only requirement for seamless cloning is the DNA inserts must share 15 base pair homology with the vector, which is P sin 6 at both ends. So here are the methods that I have used for cloning. First, PCR was used to amplify the CHS and CHI genes. And then using restriction enzyme to cut the vectors to insert the CHS and CHI genes. And then running gel electrophoresis to determine the bands with correct size. Seamless cloning was done using the GeneArt enzyme mix, and the recombinant plasmid was transformed into E. coli DH10B. Lastly, the samples were sent for sequencing to sequencing center for Sanger DNA sequencing. So this is a part of P sin 6 sequence. So the highlighted parts show where exactly CHS and CHI inserted. So we have used two restriction enzymes. One is NDE1 to insert CHI, and another one is EcoR1 to insert CHS. And this is the results from PCR. So figure A shows CHI gene with the size of 751 base pair, whereas figure B shows CHS gene with the size of 1,167 base pair. And this one is the results from vectors that I have used. So P sin 6, figure A shows P sin 6 with the size of 4.4 kilo base pair, whereas figure B shows CHI P sin 6 with the size of 5.2 kilo base pair. So this is the end product from the cloning, where we have obtained CHS, CHI P sin 6 with the size of 6,379 base pair. As you can see the map of this expression plasmid, there is neutral site, PAC origin of replication, and antibiotics, pectinomycin antibiotic, and also promoter, which is called PBSVA. And figure B shows CHS, CHI, P sin 6 band with the size of 6.3 kilo base pair. So the conclusion is one expression plasmid, which is called CHS, CHI, P sin 6, that might be compatible for gene expression in cyanobacteria, has been successfully transformed into E. coli DH10B strain. And this study will provide a better understanding on application of expression plasmid with neutral site for metabolic engineering of eukaryotic genes into microbial hosts. And here are the reference that I have used to make these presentation slides. And thank you so much for listening to my presentation. That's all from me.
Oh, thank you, Pavitra. Wow, you are early. I'm just about to come out with my bell. Okay. Well, thank you. Thank you, Pavitra. So with that, okay, so we will have our fourth oral presenter for concurrent session number 3. We have Miss Reem Abdul Salam Dawood Nadawi. Are you here with us? Yeah, Miss Reem. You may share your screen.
Yes, I'm here. Can you? Thank you very much, Associate Prof. Dr. Choi.
You may try to share your screen now.
Yes.
Okay. Full screen, please. Yeah, okay, great. Okay, so all is good. So Ms. Rim, you have 10 minutes for your presentations, and then I will ring my bell at 9 minutes and also at the end of the 10 minutes. Okay?
Okay.
So Ms. Rim is here to share with us on cyanobacteria as a source of anti-cancer compounds, evaluations of toxicity, antioxidant activities, total phenolic and flavonoid contents. So Ms. Rim, the floor is yours. Thank you.
Thank you. Very good afternoon, everyone. My name is Reem Abdussalam Dawood Nidawi. I'm a PhD student in the Faculty of Biotechnology and Biomolecular Sciences in University Putra, Malaysia. Today, I'm going to present my research with the title Cyanobacteria as a source of anti-cancer compounds, evaluation of toxicity, antioxidant activity, total phenolic and flavonoid contents. So first, a brief introduction on cancer. Cancer is a group of disease caused by unusual cell growth able to spread to different parts of the body. It's the second leading cause of death. So according to the World Health Organization, around 20 million new cases and 10 million deaths globally caused by cancer. While in Malaysia, more than 48,000 new cases and more than 29,000 deaths in 2020. So surgery and chemotherapy reduce only 5% of cancer-related death. That's why there's an urgent need for new and more effective therapies for fight this disease. Natural products are used to treat 87% of human diseases. Besides, marine microorganisms yield a vast array of phytochemicals that play essential roles in cancer prevention and cure. So here we have like a number of microalgae and cyanobacteria antioxidants. For example, we have carotenoids, flavonoids, phenolics, fatty acids, vitamin, sterols, polysaccharides, lipids, and phycobiliprotein. The use of antioxidant in cancer therapy is first to reduce the oxidative damage in the surrounding healthy tissue, also destroy the ROS scavenging capability in cancer cells, boost overall patient health and survival rates, and also decrease the cell growth, inhibit the cell proliferation, and induce apoptosis in tumor cells. So here are some examples of compounds or antioxidants extracted from microalgae and cyanobacteria that affect on cancer cells, like phycocyanin, which is extracted from Cyanobacteria spirulina platensis. It has effect on cell cycle arrest of cancer cells. We have also the beta-cryptoxanthine, which is extracted from the Cyanophora paradoxa, which affect on the which induce apoptosis in cancer cells. Oh, sorry. Also, we have another numbers like sulfated polysaccharide extracted from the Theodactylium trichornatum, which is inducing also apoptosis in cancer cells. We have also zeaxanthine, sterols, sulfated polysaccharides, and figosanthine. So what is cyanobacteria? Cyanobacteria is also known as blue-green algae. It's a photosynthetic microorganism, has different morphology like unicellular, colonial, and filamentous. It can be found in different habitats like terrestrial and aquatics, and also it can be found on the desert and hot spring environment. Why cyanobacteria? We choose cyanobacteria because it's rich in many phytochemicals and have like a wide broad of broad range of activities like antibacterial, anti-cancer, antioxidant, and antiviral. The phytonutrients and pigments present in cyanobacteria as antioxidant, which facilitate the formation of body defense mechanism against free radical damage to the cells. And also, compared to other microorganisms, cyanobacteria grow easily and survive on minimum requirement of light, CO2, and water. So the difference between bacteria and cyanobacteria is that the cyanobacteria is larger than bacteria in size. Also, it's like it contain bacteria can be contain flagella and with cyanobacteria, no. And the wall of the cell is in bacteria it contains glycolipids, while and peptidoglycan, while in cyanobacteria it's cellulose and pectin. The nutrition is usually autotrophic in cyanobacteria, while in bacteria it's autotrophic or heterotrophic. And the heterocyst is absent in bacteria, while it's present in cyanobacteria. So here we have the three types of species that we have used in this research. It's Anabaina, which is filamentous, and also it can be found in water and moist soil. Its cell range from 4 to 50 micron and contain heterocyst, which is the site of nitrogen fixation. The microcystis, which is unicellular, it's found in fresh water. It can be consist like it can produce many types of toxins like microcystis and hepatotoxin. And desertifilium, the third type is the desertifilium, which is filamentous. It can be found in hot environment like dry Thar desert in northwestern India. So the objective, moving now to the objective of this research is firstly to prepare the crude extract of Anabaina, microcystis, and desertifilium species using two solvents, methanol and chloroform, to determine the total phenolic contents and total flavonoid content using spectrophotometer. Third, to determine the antioxidant activity using DPPH and FRAP assays. Lastly, to evaluate the toxicity of cyanobacteria crude extract using the Artemia salina assay. Moving to the methodology. So here, this is the flow chart of my research. Three types of cyanobacteria extracted using two types of solvent, methanol and chloroform, to obtain the crude extract. Then to determine the phytochemicals to determine the phytochemicals for total flavonoid and total phenolics, and also the antioxidant activity using DPPH assay and FRAP assay, and to determine the cytotoxicity of the cyanobacteria crude extract using Artemia salina assay. So the first is the preparation of crude extract. We use the dry biomass and add the solvent methanol or chloroform, and then we sonicate using the sonicator at and then shaking for 24 hours, and then evaporate using the rotatory evaporate. The second step is the phytochemical screening. First one is the total phenolic content is by mixing the crude extract with the Folin-Sicalto reagent, and then add 2 ml of the sodium bicarbonate solution, incubate in the dark room, and read using the spectrophotometer. The total flavonoid contents is by mixing the extract with the aluminum chloride and sodium acetate, then add distilled water, incubate for 30 minutes, and read using the spectrophotometer. The next is the antioxidant activity. The first one is using the DPPH assay by mixing the 1 ml of the extract with the 1 ml of the DPPH reagent, incubate in dark for 30 minutes, then read using the spectrophotometer. Next is the FRAP assay. It's prepared by mixing the 20 micron of extract with the 200 micron of the FRAP solution, incubate for 20 minutes, then measure for using spectrophotometer. And lastly, is the evaluation of the cytotoxicity of the extract using the Artemia salina by using 1 gram of Artemia and adding 500 ml of fresh water, and also good aeration and continuous light for 48 hours. Then adding the 2 ml of extract with different concentration, 10, 100, and 500 microliter, and then add to number 10 of Artemia salina. Then after that, read the Artemia or the percentage of mortality of Artemia using the microscope. Moving now to the results. So here we have the yield, the percentage yield of the crude extract. We started first with 200 milligram of the biomass, and then so here we can see the final percentage yields of the cyanobacteria crude extract, which shows that the desertifilium, the methanol, the chloroform of the desert. The chloroform extract of desertifilium show the highest yield percentage, which determine that the anabina also we can be seen that the anabina, desertifilium, and microcystis show the higher yield percentage with solvent chloroform here because than methanol, because it was found that chloroform was found to be the best solvent for small volume extraction, as it's volatile and dry out faster than most solvent, because it not disrupt the bioactive compounds that are extracted and leave less contamination by plastic tube used during the evaporation. So here, moving to the
can you wrap up your presentation?
Yes. For the phytochemicals, the result of phytochemicals show that the methanolic extract of desertifilium and also the total flavonoid show that both in total flavonoid and total phenolic contents show that the methanolic extract of desertifilium is the highest in both. So for the methanolic extract of the total phenolic, the methanolic extract of anabina, microcystis, and desertifilium show the higher total phenolic content compared to chloroform, because it's believed that methanol would be better terms in extracting phenolic compounds because of the high solubility of phenolic compounds in higher polarity solvents. So moving to the antioxidant activity,
Can Ms. Rim, sorry to interrupt. Yeah, your time is up. Okay, is it okay for you to go straight to the conclusion?
Oh, this is the last. I think this is the last. Okay, just like 1 minute. Is it okay?
It's overshoot by 3 minutes already.
Oh.
2 minutes. Sorry. Yeah, please wrap it up.
Yes. For the radical scavenging activity, it shows that microcystis is the highest, while in the ferric reducing antioxidant activity, it shows that the methanolic extract of desertifilium was the highest. So here also the cytotoxicity assay, and we have using like 24 hours and 48 hours. So all of them shows that the cyanobacteria extracts for all the extract has low cytotoxicity because they are less than 50%. So they are not toxic. And specifically, desertifilium species of methanol was the lowest toxicity. So in conclusion, the results show that the methanolic extract of desertifilium contains the higher amount of total phenolic contents and also total flavonoid contents with the 90, and also it was the highest antioxidant activity through FRAP assay, while the microcystis show the highest antioxidant potential through DBPH assay. Besides, all the extracts show low toxicity against Artemia salina. In conclusion, this study revealed that desertifilium is an excellent source of natural products from cyanobacteria, which could be explored further for its potential in becoming a new anti-cancer treatment, as it has low toxicity and high total phenolic phenolic, total flavonoid, and antioxidant activity. Thank you for your the acknowledgement. Also, I would
Thank
like
you,
to
Ms.
express my
Lim.
deepest appreciation. And thank you. This is the
Okay.
reference, and thank you for your attention.
Okay, thank you, Ms. Ring. Okay, thank you. Okay, we have our fifth oral presenter today, 019, Ms. Noor Azima Tajuddin. Are you here with us? Very good, thank you, Ms. Noor Azima. Yeah, I can see your screen.
Yeah.
Can you put it up for full screen?
All right. All right. So can you hear me? Can anybody hear me?
Yes, loud and clear. Okay. Yeah, due to the time, I guess, okay, I will leave the title to you. Ms. Noor Azima Tajuddin is from UITM Sungai Buloh. Okay, so the floor is yours.
Right. Assalamualaikum. Thank you. First of all, thank you to Prof. Dr. Choi. My name is Noor Azima Tajuddin. I'm a postgraduate student from ICMBB UITM Sungai Buloh. So today I want to share my part of research project, which the title is Identification of SARS-CoV-2 Omicron variant by Oxford Nanopore sequencing in a tertiary institution, Malaysia. So this is my presentation outline. I will go one by one later on. As for SARS-CoV-2, let me refresh back. So the SARS-CoV-2 virus is globally taken numerous cases in the world, including Malaysia, since it was originally detected in Wuhan province, China, back in 2019. Up to April 2023, more than 672 million confirmed cases of COVID-19, which include more than 6 million that was reported worldwide. And from February 2020 to the end of 2021, Malaysia already experienced the global pandemic SARS-CoV-2, including the variant of concern Alpha, Beta, and also Delta. And now Malaysia was entering the transition phase to endemicity since last year, April. So during that time, testing of COVID-19 now was mainly done with rapid test kit, and also surveillance also carry on in this country. So talking about Omicron was first detected in Malaysia back in November 2021, and it's continuously circulating until now. As for now, Omicron subvariant XBB were most circulating in Malaysia since last year, October 2022. Last month, on May 2023, WHO already announced the COVID-19 as established an ongoing health issue which no longer constitute a public health emergency of international concern. So it means that it was no longer public threat, and people already vaccinated. The coverage of immunization in Malaysia itself already occupied more than 85%. And people also know how to isolate themselves if they are infected with COVID-19. Hospital UITM Puncak Alam was actually located in Selangor, Malaysia. It's the tertiary hospital which also received cases of COVID-19. This hospital later renamed as Hospital Sultan Abdullah in the officiating ceremony last year, July. And this sequencing platform provide real-time sequence information and also can help in clinical diagnosis in hospital setting, as well as management of contact tracing. So my project aimed to use this nanopore sequencing to identify Omicron variant in patients admitted in this hospital. So why actually I chose the Oxford Nanopore sequencing technology? It was this sequencing, a third generation sequencing, which offer fast, reliable, and also accurate result. And this nanopore use flow cell, which contain nanopores, which the current is applied to it. And this nanopore will allow the RNA molecule pass through the membrane by presence of electrical current and buffer. So when a molecule passes through a nanopore, the current is then disrupted and the sequence is generated real time. So the advantages, of course, it was less laborious. It only require minimal lab setting. It also cheaper compared to the first and second generation sequence. It can sequence single molecule of RNA or DNA directly, and of course, it will provide fast result of the sequence information. So this is the method that I do in my project. So I take 53 respiratory sample from the oropharyngeal swab and also nasopharyngeal swab in the BTM from the tertiary hospitals. And I perform 3 steps, which is first is the pre-library step, second is library preparation, and also third is sequencing itself. So for the first step, I do the normal RNA extraction and also PCR amplification. And then the product then will convert into cDNA and use the rapid barcoding code in the library preparation. And then the library was loading into the sequencer. So I want to highlight the library preparation method, which I use the rapid barcoding kit. They have 5 steps here. So it will start from the cDNA conversion, and then I amplify with ignite primer, and then the product will barcode using the specific barcode. And after that, the pooling will take place, and I will clean up all the remaining residue with using the MPO bits. And before loading to the flow cell, we'll prime and also wash the flow cell first. So all these 5 methods is approximately taking about 5 to 6 hours depending on the sample. So more sample needs more time because we need to do more pipetting technique in this step. Okay, go to the nanopore sequencing. So nanopore sequencing start when you load the library inside the nanopore, inside the sequencer. For this one, I use the MK1C sequencer, and then it will run for about 16 to 18 hours, depending on for the sample. And the software inside this sequencer will analyze the result and will perform the local base calling, post-run barcoding, and also alignment, and assembly and also variant calling. So it will provide the final result as raw sequence in FASTQ input. So I will use the FASTQ input into the epitomy analysis in order to do simple bioinformatic analysis and reporting. So this is the result that I get from my sequencer. I will upload all these results in the FASTQ form into the epitomy desktop agent, which is the simple bioinformatic software. When I get 100% coverage of result, so this bioinformatic software will do will provide the data in the variance VCF format and also consensus in the master format. So this is example. This is the result that I get from the epitomy. Also integrate with the NextClick QC. So what NextClick QC do? NextClick QC showing the visualize the genetic variant and also assist in the data quality control. So from here, I can get what is actually the variant, what is the lineage, the clade, and also I can see if the sample is in good sequence or not. And this is showing the mutation on the spike protein gene. So this is the example of the mutation. And beside the NextClick QC, the result also shown in the pangolin lineage assigner. Also, it can show what is lineage that I get from each barcode of the samples. So I tabulate all the data, and then I do a simple tabulation in this table. So from my 53 sample, I get all Omicron, which is 13 of them is from BA1 and BA1.1 and also BA1.17. And then another 40 sample, I get BA2, BA2.3, and BA2.9. So it can show what is the pangolin lineage, what is the clade of the Omicron, and what are the one of sample isolated. And this is I show the mutation that I found in the spike protein throughout the analysis of the NextClick. So from this result, I tabulate into the phylogenetic tree. So this is the circle shape of phylogenetic tree of SARS-CoV-2 Omicron variant that I isolate from this tertiary institution. I construct using the neighborhood joining method in Mega7 software. So from 53 sample, I can get the BA1.1 and BA1.7 were detected in February and March 2020, and then followed by BA2, BA2.3, and also BA2.10 throughout the February until June. So this finding is similar with the other country where the SARS-CoV-2 Omicron variant are diverse within the same time frame, which is the first 6 months of 2022. So in conclusion, sequencing now form the base of biology research, and it was applied in medical diagnosis as well as biotechnology, forensic biology, as well as virology. It offers simple and also less laborious method in identifying variant of SARS-CoV-2. And this nanocode sequencing provides fast and real-time result. I can use the simple bioinformatic pipeline to generate all the result using simple and also I can get real-time support from the nanopore community. So don't worry, if you want to do the nanopore sequencing, you always can get the support from the nanopore. From my result, I can conclude that BA2 sublineage has been outcompeting previously dominant BA1 as the result of rapid transmission within 6 months time in the hospital in Shah Alam. And also impact of sequencing in clinical diagnosis, it can be used in contact tracing and also help the public health measure. And lastly, sequencing is crucial in understanding more about genome level, and also it can identify and describe new mutations. So this information is very resourceful for scientists or researchers that want to study more about the genome organization. I think that's all for my presentation. For that, I thank for my lab mates, my supervisor, Dr. Siti Farah Alwani, and Associate Professor Dr. Wang. IMMB staff, Faculty of Medicine. I promise you, ITM Putra Alam, also National Public Health Sungai Buloh for their guidance and also support me throughout this project. Till then, thank you.
Thank you. Thank you, Ms. Norazima. Just on time. Okay. Thank you. Interesting project. So we will move on to our sixth oral presenter for this session, O20. Okay, we have Ms. Susanna Adnan. Can you share your screen?
Okay, test. Can you see the slide? Test. Hello.
Yeah, we can see the slide.
Okay, thank you. Where's Doctor? Can I start?
Yes, please.
Okay, thank you. Okay, Assalamualaikum and good afternoon. My name is Susanna Adnan from University Malaysia, University Pendidikan Sultan Idris, UPSI. So today I'm going to present my research finding entitled Cloning, Expression and Purification of Recombinant Multicopper Oxidase lacase from Pseudomonas putidum-4. Okay, basically my research will be about extracting a protein or enzyme named multicopper oxidase lacase from a bacteria named Pseudomonas putidum-4. This lacase was functioned in degrading polyaromatic hydrocarbons, which is hazardous, to form less hazardous forms of PAH and water as their side product. Okay, what is polyaromatic hydrocarbon? Polyaromatic hydrocarbon is a class of organic compounds that is composed of multiple aromatic rings. So this is the example of polyaromatic hydrocarbon, which is naphthalene, anthracene, and phenanthrene with different amount of benzene rings and benzene composition. Okay, the source of polyaromatic hydrocarbon can be from crude oil, cigarettes, burning of woods, and even in our food. If we cook our food, overcook the food, it can lead to production of PAH also. Okay, research has been done in Malaysia about the distribution, sources, and ecological risk assessment of PAH in Malaysia. For example, in Johor Strait, Klang Strait, Kuala Lumpur, Port Dickson, Kapas Island, and Langkawi, Kedah. Okay, and Chu and friends reported that there are three highest concentration of PAH found in surface sediment of Kapas Island, which is naphthalene, phenanthrene, and anthracene. Okay, let's throw back to what happened, the incident in our country during 2019, where thousands of people affected by inhaling a very toxic and dangerous fuel gas from Sungai Kim Kim River in Pasir Gudang. Okay, long-term exposure may decrease the immune function, cataract, kidney and liver damage, breathing problem, asthma-like symptom, and lung function abnormalities. Okay, when pH pollution occurs, usually the method to clean up the pollution is that by photolysis, volatilization, adsorption, chemical degradation, and bioremediation. For my research, I focus on bioremediation. Bioremediation is introducing microorganism that has the capability to degrade organic compounds to produce less harmless substances such as carbon dioxide and water as their side product. And this is the example of microorganism that have been used in bioremediation, which is Pseudomonas aeruginosa, Pseudomonas fluorescens, Mycobacterium, and etc. Okay, a part of bioremediation is bioaugmentation. Bioaugmentation is in situ introduction of the microorganism into the polluted area. Okay, but Pete Soto and friends has reported that bacteria itself could not survive for too long and lead to death phase. Okay, one of the solution to this weakness method is that we will introduce lactase or other enzyme that more stable and strong activity in this extreme condition. Okay, what is lactase? Lactase is a multi-copper oxidase, MCO, that catalyzes the oxidation one of electron of a wide range of phenolic compounds, and it yields water as a sole byproduct. Okay, this is the flow for my research methodology, which is first I will do DNA extraction, and then I will run PCR, gene cloning, protein expression, and protein purification. Okay, first DNA extraction, we will incubate culture for overnight, and then we harvest the cell on the next day, and we lyse the cell to get the DNA for the PCR. Okay, this is the result for the PCR. We design the primer and we run the PCR to get our targeted gene, which is multi-copper oxidase gene. So we managed to get the band with the right size here, which is 738 base pair. And then to confirm the band that we get from this PCR product is multi-copper oxidase, so we send the PCR product to sequencing, and then from the sequencing result, we blast the result at the NCBI gene bank, and then we get up to 88.7% similarity of the protein sequence with the multi-copper oxidase from Pseudomonas putida strain CA3, Pseudomonas putida strain KT2440, and Pseudomonas putida strain NCIM 2650. Okay, once we confirm that the PCR product is our targeted gene, so we proceed with gene cloning. Okay, in gene cloning, we clone the gene into the PJEX41 vector and E. coli BL21 DE3 as the host. Okay, this is the result from plasmid extraction to confirm that our recombinant plasmid contain our targeted genes. So here we can see the correct size of vector and the correct size of our targeted genes, which is multi-copper oxidase gene. Okay, so once we've managed to clone the recombinant gene into the E. coli, and then we express the protein by using IPTG. Okay, this is the result for the STS page. As we can see here, the size of multi-copper oxidase, which is 54 kilodalton, compared to the control, there's no band here compared to the induced protein here. Okay, next, to determine the function, to make sure to know whether the protein itself has the activity, Lackey's activity, we run 3 types of assay, which is anthracene assay, glycol assay, and ABTase assay. And these 3 assays shows positive results. Okay, and then we move to the protein purification steps. And in this step, we use a glutathione sulfurous phosphorescine. And then this is the chromatogram flow for my purification step. And then this is the STS page result for my purification step. And we managed to get, I can say this is 90% purified multi-copper oxidase gene from here. You can see here with the correct size. Okay, this is my purification table for multi-copper oxidase. As you can see here, the enzyme purity fold increased from 1 to 3.6, and specific activity increased from 24 unit per milligram to 87 unit per milligram. Okay, as this discussion, 54 kilodalton intracellular multi-copper oxidase lacase was successfully expressed and had positive activity when tested using 3 different substrates, which is anthracene assay, glycol assay, and ABTase assay, and only require a single-step purification method, which is affinity chromatography, to receive more than 90% purity with 3.6-fold increase in enzyme activity. As the conclusion, lacase purification can be further optimized for a better expression in future, and in-depth studies about lacase characteristics, for example, stability of the lacase in pH, temperature, organic solvent, and etc., are important to produce an excellent lacase that can withstand its function in extreme conditions. With that, thank you.
Okay, thank you, Ms. Susanna. My apologies for the line drop. My line was dropped just now, and you have started the presentation. Thank you. Yeah, so I think due to the time constraint, let's move on to our last presenter for this session, O21. Okay, Ms. Halimatun Saidia Abdul Wahab. Are you here with us?
Yes, Prof. I'm here.
Yeah, Ms. Susanna, you might mute and yeah, thank you.
Okay.
I can't see you. Have you turn on your camera? Yeah, okay, thank you. Ms. Halimatun, can you share your slide? Screen. Yeah, I think it's good. Okay. Yeah, we can see your slides.
Yeah.
Yes. Yeah. Okay. So now we have Halimatun with us this afternoon. She's from University Technology Mara, Malaysia, and her presentation today is entitled Combinations Treatments of Chitosan Loaded Thymoquinone Nanoparticles and Hyperthermia Reduced, the Size and Cell Viability of Osteosarcoma Sporoids. Okay, so the floor is yours, Ms. Halimatun, and you have 10 minutes. Thank you.
Okay, hello everyone. So as what Prof said just now, my presentation is based on combination treatment of chitosan loaded thymoquinone nanoparticles and hyperthermia reduce the size and cell viability of osteosarcoma spheroids. So let's start with the table of contents. It consists of this introduction, problem statement, objective, method, results, discussion, and also the conclusions. Okay, this is, let's go to the introduction. Let's talk about osteosarcoma. So osteosarcoma is the highest cancer-related death in pediatric population and the second highest among older adults, which is more than 60 years. So according to Aziza et al., in their study, they reported that 858 new cases were diagnosed between year 2007 to 2011, and another 910 cases between year 2012 and 2016. The challenges in treating this malignant are due to their heterogeneity and aggressiveness that makes the treatment is more challenging. So despite the limb sparing, surgery, amputation, chemotherapy, and radiotherapy, the 5-year survival rate for localized and metastatic osteosarcoma remains stagnant, which is at 52% and 22% respectively. So this is due to the chemoresistance to drugs and also the lack of targeted therapy that can fully eradicate the cancer cells. Okay. So this is the osteosarcoma at the epiphyseal plate. As we can see here, this is the epiphyseal plate. This is where osteosarcoma started to form. Okay. So what is thymoquinone? So thymoquinone is 2-isopropyl 5-methylbenzo-1,4-quinone, which is the main active compound in Nigella sativa. So Nigella sativa exhibit antiproliferative effects on cell lines which derive from breast, colon, ovary, larynx, lung, myeloblastic leukemia, and also osteosarcoma. According to Sarman et al., in their study, concluded that when thymoquinone combined with low doses for 5-fluorouracils on oxaliplatin, it inhibit resistance towards 5-fluoresceins and oxaliplatin, which is the cell viability decrease and apoptosis increase. The thymoquinone also can elevate growth inhibition and apoptosis, which reported that when tested on human osteosarcoma cell line as AOS2. So according to Al-Matrodi, and this situ cells viability after treated with thymoquinone was reduced compared to normal human osteoblast cells, which means thymoquinone is less cytotoxic to normal cells, but it's cytotoxic to tumor cells. So thymoquinone also induced cytotoxicity, which was seen in human colorectal cancer cell lines, CACO, LOVO, HCT116, HT29, and DLD1, but not in normal human intestinal cell line. So cytosine nanoparticles thymoquinone need to be encapsulated in a carrier for it to be delivered effectively to the core of the cells. So, we in this study we encapsulate thymoquinone in cytosine nanoparticles, which is the cytosine is water soluble polymers which obtained from chitin, which is a natural polysaccharide composed of beta-1,4-link and acetylglucosamine units. So cytosine, it possesses excellent biological properties as it is non-toxic, mucoadhesive, hemocompatible, biodegradable, and possessing anti-tumor, antioxidant, and antimicrobial properties. It also is excellent carrier as it has better stability, low toxicity, simple, and it needs mild preparation method and allow versatile routes of administration. So cytosine nanoparticles reported to be toxic on A549 lung cancers, but not in normal cells. So the chemotherapeutic agents loaded with cytosine nanoparticles showed anti-cancer potency with reduced adverse effect compared to the drugs alone. So cytosine loaded thymoquinone nanoparticles. So it has lower dosage of TQ needed to kill MCF7 cells by 50% when encapsulated in CNP compared to free TQ, free thymoquinone. Study by Alam S showed that intranasal cytosine nanoparticles thymoquinone more effective in brain targeting as compared to free thymoquinone solution delivered by either intravenous or intranasal in Alzheimer's study. So a study by Guinea showed that thymoquinone when loaded in PLGA, which is polydyl L-lactate glycolide, showed effectiveness in inhibiting the growth of breast epithelial cancer cells compared to the free thymoquinone. This is the chitosan nanoparticles which are encapsulated in thymoquinone, modified with L-ascorbic acid. So, hyperthermia, chitosan have good thermal stability and can withstand temperature up to 200 degrees Celsius. And according to Aisha et al., heating osteosarcoma MD63 cells for 45 degrees Celsius for 1 hour lead to induction, double-strand DNA damage, and apoptosis. The significant reduction in cell growth of osteosarcoma cells after hyperthermia treated at 42 degrees Celsius also found by Treat et al. So, according to Jose et al., moderate temperature of 40 degrees Celsius to 45 degrees Celsius can kill or induce tumor cell apoptosis without damaging the normal cells. So, hyperthermia can also induce the endoplasmic reticulum, triggered apoptosis in numerous cancer, osteosarcoma, melanoma, skin cancer, colon cancer, lung cancer, and also breast cancer. So this is a problem statement. The resistance to chemotherapy drugs and lack of targeted therapy are the biggest challenge in osteosarcoma treatment. The combination of thymoquinone nanoparticles and hyperthermia leads to a new breakthrough and resolving the issue and provide better quality of life among osteosarcoma patients. This is the objective. The main objective is to examine the combination efficacy of chitosan nanoparticles containing L-ascorbic acid and thymoquinone and hyperthermia in modulating MD63 cell viability and sporide site following first and second recovery phases. The specific objective consists of two, which are the first one is to investigate the effect of CNP-LLATQ alone and combination chitosan nanoparticles loaded with thymoquinone and hyperthermia on osteosarcoma MD63 spheroids viability. The second one is to measure the size of MD63 spheroids after treatment with chitosan nanoparticles, thymoquinone, and combination with hyperthermia following recovery phase. So this is our methodology. The cell cytotoxicity on chitosan thymoquinone, the chitosan loaded thymoquinone nanoparticles were done, and we found that we treat with several concentration at 4,500 to 7,500 microgram for 48 hours. And then the SSA was performed. We found that at 5,000 microgram, the cells were killed by 50%. So using the IC50, we treat the cells for 48 hours using thymoquinone PLGA. This one is control group and treatment group. So this one, we undergo hyperthermia for 1 hour and then undergo recovery for another 24 hours. This is the first stage recovery. And then this all, we will perform the cell proliferation using TRIPA-BLUE, spheroids measuring image, using J software. And we also do cell viability test using MTS assay. The second one is using the same concentration, we treat for another recovery, another phase for 48 hours. Then we treat for hyperthermia after 48 hours for 1 hour at 45 degrees Celsius, and then undergo recovery for another 24 hours. So after that, we undergo this all three tests: cell proliferation, spheroids measuring using image software, and also cell viability using MTS assay. So this is the result. This is the first phase recovery result, the control group. As we can see in control group, the spheroids become bigger and more dense from day 3 to day 6. For treatment group, the spheroids become shrinks and less dense and looks like they start to die and it shrinks until day 6. The diameter also decreased. There's also significant decrease in diameter, in cell density, and also cell density. So this is the second phase recovery. This is the control group. Day 2, day 0, day 3, day 5, day 6, day 8, and day 9. So as we can see, the diameter increased and the density also increased in control group. For treatment group, yeah.
Hi, Miss Halimatun. Sorry to interrupt. Sit up. Can you wrap it up?
Okay. So this is the treatment group for second phase recovery. So the cells look very shrink and die after day 9. So this is the spheroids viability, the first recovery and second recovery. So this is the discussion. As we can see, using the spotting method, MDCT cells were seeded at 20,000 cell density in 96 wells with cell gas permeable plate. After the spheroids formed on the tree, cytosine loaded thymoquinone nanoparticles of 5,000 microgram per mil concentration was used to treat them for 40 hours, followed by 1 hour hypothermia and 24 hours recovery. And there's another set of spheroids in 96 well plate were treated with the same cytosine loaded thymoquinone nanoparticles concentration for 40 hours, followed by 1 hour hypothermia and another 24 hours recovery. Then the spheroids were treated again with the same concentration of cytosine loaded thymoquinone nanoparticles for 40 hours, followed by 1 hour hypothermia and another 24 hours recovery. MD6CG spheroid size was significantly reduced after combination treatment with hypothermia during second treatment compared to the first treatment. The cell's viability of the spheroids are also significantly decreased to 28% in first phase treatment, while in second phase, the cell viability reduced to 22%. Okay, so the conclusion is cytosine loaded thymoquinone nanoparticles with combination of hypothermia are dose and time dependent as exposure to MD63 to cytosine loaded thymoquinone nanoparticles and sensitize and kill some of the spheroids during first treatment and kill cells more effectively in second treatment. So these are my references and thank you, that's all.
Okay, thank you, Ms. Halimatun. So we are actually running out of time. It's already 15:25. Okay, so we do have one question here from Dr. Dr. Let me scroll up. From Dr. Chong Fei Wen. The question is directed to Miss Susanna. Okay, so this is regarding the blast results on your MCO genes. Okay, all of them show lower than 90%. Okay, so Dr. Chong is wondering whether did you look into the detail of your alignment? Miss Susanna?
Yes.
Oh, I saw your yeah. Please.
Yeah, I do reply at the chat box here, but okay. Yes, for the explanation is that I do run the detailed alignment between these 4 sequence, and it has difference at the nucleotide sequence. But when I translate into the protein sequence, it is actually 100% similarity between these 4 protein sequence. Is it accepted? Okay.
Okay. I guess probably you may want to discuss with Dr. Chong on the questions, but I do need to raise here is where you know the difference between similarity and identity, right? Okay. So please take note on the similarity and identity is actually different. Okay. So I guess I do have some questions for our participants here, but I think due to our limited time, I just wanted to pose one questions to all the oral presenter this afternoon, okay, from 015 to 021, okay? So what is the challenge, okay, of throughout your research? Okay, what is the challenge? Maybe one of you want to share with all of us here the most challenging thing that you have seen through in your research up to this stage. Anyone?
Can I
Anyone?
respond?
Yes, sure. Thank you.
Okay. For my case, the most challenging part is when I do molecular works, which is involve DNA, RNA. The most challenging part is to maintain the sample itself in the freezer. When the light electric cut happen in the building, then the sample is gone, and it track my research. Basically, yeah, that's the most challenging part. Okay, thank you.
Okay. Okay, thank you, Miss Susanna. Okay, I think we have to wrap up. The time is up. So with that, let's put our hand together. Thank you for all our 7 oral presenter for these sessions. Thank you for the presentations, and we shall move on to our next session, which is our poster presentation. Okay, back to the technical team. Thank you.